Let us go ahead and get started. So welcome everyone. This is a tutorial, a pre-CNS tutorial about the Allen Institute data, tools and models. It's great to have you here with us. I'm Anton Arkhipov and I'm here together with several other colleagues from the Allen Institute. We will now do a quick introduction and then we'll be on our way with talking about, well, the data, tools and models. So first things first, this is a webinar, right? And so we love all the attendees to use chat and Q&A. Just use them for slightly different purposes. So for the chat, we ask you to use it for general conversation. And now might be a good time to introduce yourself. Please write down your name and who you are. It's great to have you all here. And so, yeah, just keep an eye on who you are sending the message to. It might be to all panelists or to all panelists and attendees. In that case, everyone will see it. And again, chat is just for general conversation. Try not to post your questions there because they might get lost and it might be hard to keep track of them. Instead, please post your questions in Q&A, questions for speakers. It can be also questions for the group as a whole, including other attendees, requests for clarifications, all of that. If you could post in Q&A, that will be great and will be easier to keep track of. For all of the tutorials that we have today, we will we are trying to leave some time at the end of each tutorial, and each of them is an hour long. So we'll try to leave some time at the end for the speaker to answer all the questions that accumulated. But if there is something urgent or very important question that requires a clarification during the presentation, the speaker may choose to stop and answer it right away. But otherwise, we'll try to answer them at the end. All right, so I hope this is all clear. And yeah, it will be great to hear your questions and introductions. And now I'd like to take just a few minutes to tell you a little bit about who we are and what we'll be talking about today and tomorrow. So this group of speakers, we are from the Allen Institute, which is in beautiful Seattle. It's an institute that, as many of you know, was established in 2003 by philanthropist Paul G. Allen. It's in the South Lake Union neighborhood of Seattle, where all the other high-tech establishments are. It has over 500 employees now. And also importantly, we are doing a number of different, we are pursuing a number of different directions of research, not only in neuroscience or brain science, but also in studying cells, in immunology. There is also a Frontiers group that does intramural funding. And the overall direction of work for all of these groups, especially for us in-house in the institute itself, is to tackle hard problems, complexity, foundational questions in biology with the approach of big science, team science, and open science. And that results in data, as well as knowledge and tools, which we share freely with the community. So a few words about these core principles here. This is really important for how we operate. And this is done in a sort of as a complementary approach to the very successful approach in the university labs. So now, unlike in university labs, instead of pursuing individual sort of focused research projects, we try to work as big teams in this interdisciplinary approach with multiple teams working toward common goal. So that's the team science component. And that brings about the second component, which is big science. So with this team approach, we are able to tackle large scale projects that are too big, too complex for university labs, or typically for university environment. So we are usually joining forces of multiple groups to provide a big data set that's as complete as possible and with very high quality standards which we then share with the community. And so that brings us to the third part, which is the open science. And that's really the key to the way we operate at the Institute. We make all our data, all our tools available to the community on our website, brainmap.org, or on GitHub if we are talking about the software tools. And typically we do that 1 to 3 years before any publication. So again, this is really important piece for us to share everything freely with the community. All right, so now the Institute started in 2003, and it started with producing a number of data sets that became quite popular in the field, especially the Allen Mouse Brain Atlas of gene expression in the mouse brain, and a number of others. In a recent decade or so, we sort of branched out into this more systems neuroscience type of work, producing more new data sets, which are highlighted here. So we have data sets about cell types in the mouse and human brain. We have the Brain Observatory, which is the recordings of activity of neurons in the mouse cortex and other structures. Using two-photon physiology, as well as more recently electrophysiology using Neuropixels probes. We have models, we have multi-patch synaptic physiology and electron microscopy characterization of connectivity. And the latest addition is this visual behavior optical physiology data set, which is the in vivo activity of neurons of different types from the mouse cortex in a context of a particular behavioral task. So all of that and actually more than this is available online. And today we are going to cover several of these data sets. Actually, today and tomorrow we are going to cover several of these data sets that are shown marked by a star here, right? So we'll talk about cell types, we'll talk about synapses, we'll talk about activity in vivo using optical two-photon physiology, calcium imaging, and electrophysiology with Neuropixels in passive viewing and in behavior, and we'll talk about models. All right, and now also just a few words about the structure at the Allen Institute. This is something that a lot of people are not aware of, but we actually have multiple organizations within the Allen Institute, even in the area of brain science. So one of them is what is called the Allen Institute for Brain Science. And today and tomorrow, there will be representatives of this organization here. So the examples of what the Allen Institute for Brain Science does are shown here. These are the studies of the structure of the brain. So cell types, what the brain consists of, right? Using the transcriptomics, morphological characterization, electrophysiological characterization. Connectivity, how these different cells and cell types are connected with each other using multi-patch synaptic physiology or using electron microscopy, as well as other approaches. And then something that's not shown here are other approaches and data or tools that are really available in the community. For example, the transgenic mice, which are shared freely and are very popular for the experimental work. So many different facets of work in this organization, and we will hear from the representatives of this organization today and tomorrow. And then the other organization that also does research in the brain science is called the Mindscope program, the Mindscope program at the Allen Institute. And the work in this organization has to do more with activity in vivo and sort of the computations that the brain cells perform. So a big part of this program are the experiments in behaving or passively sensing mice and recordings of neural activity using electrophysiology, as I said already, with the Neuropixels probes, or optical physiology with calcium imaging. And then on the other hand, we also have an effort in this organization that does modeling at different levels of resolution. And today and tomorrow we will hear about all of these aspects from the representatives of the Mindscope program. All right, and so this is our schedule for today. So we are almost done with the introduction. Then next will be Luke, who will tell us about the synaptic physiology data set. Then we will have a short break. And the remaining 2 tutorials today will be by Saskia and Josh about visual coding using 2-photon calcium imaging or Neuropixels recordings in vivo. And tomorrow there is another exciting day. So we will have Nathan speaking about cell types and characterization of transcriptomics, morphology, and electrophysiology in different cell types using the patch-seq technique. Then there will be a break, and the visual behavior data set will be described by Alex. And then we'll conclude with talking about the biorealistic modeling. So we'll hope to see you tomorrow here as well. Finally, all the tutorial materials are available here on this webpage on the GitHub Allen Institute CNS 2021. So feel free to go there and follow along in the Jupyter notebooks with the tutorials that we'll go over today and tomorrow, or just keep it there for your information, and feel free to download later and play around with these notebooks, but they are there for you on this webpage. Okay, so with that, I'll finish and pass it on to Luke to tell us about the synaptic physiology data set.
Thanks, Anton. So before I start, I'm going to post a link in the chat to, this is a collab link to the notebook that I'm working on. So if you click that link, you can follow along and run those notebook cells as we go. It does require a Google account. So sorry if that doesn't work for you. We can help you get it set up later on. So as Anton said, my name is Luke Campagnola. I do research with the Synaptic Physiology group at the Allen Institute for Brain Science. I'm going to talk today about a project that we've been working on for several years. Maybe I should share my screen for you. So we've been looking at the electrophysiological properties of synapses in mouse and human cortex. And the main question here is, what are the relationships between cell types and synapse properties? So the term cell types is not very well defined. So to be a little bit more clear, I'm talking about pretty broad classes of neuron defined by their location in the cortex, which layers they're in, but defined by their morphology, and specifically whether we can tell if the cell is excitatory or inhibitory just from their morphology. And also any transgenic reporters that we've introduced into tissue. So for example, in mouse tissue, a typical kind of cell categorization we might do is say like layer 5 somatostatin neurons or layer 2-3 pyramidal cells, things along those lines. And likewise, we do this work in human cortical tissue that's come from surgical resections. And we don't have access to transgenic reporters in those cases, but we do still use morphology and layer, as well as electrophysiological properties. So to give you an example of the kinds of questions we're looking at here, what is the probability of finding a synaptic connection between 2 cells? You pick any 2 cells in the cortex. What's the probability that they're connected by a synapse? And how is that probability affected by the cell types that you're looking at? How is that connectivity affected by the distance between the 2 cells or by any other experimental parameters? How is cell type related to other synapse properties, such as strength and the latency and the kinetics and the dynamics? And how can we best model synaptic connectivity and dynamics? And what can these models tell us about the function of the circuit? So that's sort of where we're headed with this project. The technique is patch clamp electrophysiology. We use multiple electrodes at the same time. So we will acquire a very thin slice of brain tissue, typically about 1/3 of a millimeter thick. This is live brain tissue. We bathe it in artificial cerebrospinal fluid to keep it alive for several hours. And we use these glass electrodes pictured here to attach to individual neurons, and that gives us electrical access to the interior of the cell. That gives us two things. First, it allows us to stimulate the cell to fire action potentials very precisely, and it allows us to record from other neurons at the same time. So the idea is one cell fires an action potential, and if the next cell happens to be connected by a synapse, we should be able to see the resulting currents from that synapse. In a single experiment, we typically patch up to 8 neurons simultaneously. So we will go around each cell one at a time, firing action potentials and listening in all of the other cells. And in mice, what I have pictured here is we often have fluorescent reporters. So we will typically look for 2 different classes of neuron at a time. So for example, here, the red cells might be labeled for somatostatin cells, and the green cells are maybe a layer 5 pyramidal cell or something like that. So once we locate a synapse, once we've actually found a connection, our next job is to characterize the synapse. And so that means stimulating presynaptically with many action potentials in many temporal patterns. And we're going to average together all of the synaptic responses to get parameters like the amplitude and the variance in the amplitude and the shape, the rise, time, and decay, and the latency. So how long was it from the presynaptic spike to the onset of the postsynaptic response? And then in addition, we force the presynaptic cell to fire in very rapid trains. And that allows us to see what happens to the strength of the synapse during rapid stimulation. And it's pretty common to see synapses that either facilitate, which means that their amplitude gets larger, or depress, their amplitude gets smaller. And there are many other combinations and exceptions to those. So once we've collected all of that data, it heads into a database. These are downloadable database files. It's just a relational database. It's an SQLite file. It's spread out over many tables, as you can see here. But we're going to show you the simplest way to access the data. But to give you an idea of how this is laid out, we have 3 main categories of data that are here. These 2 tables on the left contain experiment metadata, things like the species, the brain region, any experimental conditions. And then we've got a few tables here that describe cell properties. So that's individual cells, their morphology, their intrinsic electrophysiology, and their location within the cortex. And then the rest of the database is devoted to synapse properties. So in the center here, we have this table called pair, and each row in this table, each record represents 2 cells, one presynaptic cell and one postsynaptic cell. So this is sort of the central point in the database, where we link together information about cell properties and synapse properties. So in order to query information, the vast majority of the time, we're going to be querying directly from the pair table and then attaching data from cell and synapse and from experiment. So if you're running on Colab, the first thing you need to do is install these 3 packages just by hitting this play button. You'll get a warning here that says this was not authored by Google, and it means that the code that you're running has access to some amount of data. I actually don't know how much data of yours it has access to. You can either hit cancel and just watch my presentation, or trust that none of my code is written to look at your data. So right now what we're installing is this package AI SynPhys, which is tools that we have built for accessing the database that we just saw. Neuroanalysis is supporting functions for analysis that we do in the background. And LMFit is just a curve fitting tool. These are the only 3 dependencies that were not already present in Colab. All right, so now, first thing I want to do is load up one database file, and that's happening on this line right here. I'm saying load current, and small means I want the small version of the database. Our databases come in 3 different sizes: a small, medium, and a full size. For everything we're doing today, the small is totally adequate, and it's going to download much faster. So I'm going to run this. And immediately after I get this database right here, I'm going to create a query. And this is a pair query, which means I'm asking for records out of that pair table. And I have a bunch of filters specified here. So I'm saying experiment type equals standard multipatch. This appears in every query in this notebook, just because we have a variety of different types of experiments in the database. And for now, all we want are these standard experiments. I'm restricting this search to all human data. I'm restricting the search to pairs that are connected by a synapse, and specifically where the synapse is excitatory. And this last line says that I would like to also load up data about the cells, the pre and postsynaptic cells for each pair, as well as synapse properties. And finally, I ask for a data frame. This creates a pandas data frame from the query results. And you can see we've loaded 376 synapses, and the resulting data frame here is 363 columns long. So there's a lot to dig through here. Many different synapse and cell features that might be of use. All of these are documented here in our documentation. Let's just have a quick look at that. So this is the documentation for our database schema, and you can see that there are many, many tables here. The diagram I showed you earlier was only a small fraction of our tables. And as an example, here's the gap junction table, and this contains information about electrical synapses that we found. So if you want to dig into the data, this is the place to go to figure out what exactly is in the database. But for now, I want to bring your attention to just a small set of fields that are in this data frame that we just created. These contain things like the PSP amplitude, which is the median amplitude of the synaptic responses. This is basically a measure of the synaptic strength. We have latency, the kinetic parameters that we just discussed earlier. STP induction is about short-term plasticity induction. So this tells us whether the synapse is facilitating or depressing. And we have variability, which tells us about the coefficient of variance of the amplitude. So as a quick example, let's just get a histogram of all of the PSP amplitude values. And that's here. And as you can see, everything is positive. That's good because I requested in my query that I wanted excitatory synapses. If there were inhibitory synapses in here, they would show up on the negative side. You can see that most of these synapses in the human data are around 0.5 millivolt to 2 millivolts, and a very few of them go up much larger. So now we have all of this data. We have many, many synapse properties in this data frame, and our next job is to categorize those synapses based on their pre or postsynaptic cell properties, and then make a comparison. So there are a whole lot of cell selection criteria for us to pick from. We've already picked human synapses, so that limits our cells to a fairly small chunk. We could pick cells based on their cortical layer. based on their intrinsic electrophysiological features or based on their morphology, specifically whether they're excitatory or inhibitory. So again, we could go to the documentation to figure out what all of these fields mean and decide what to filter on. But there are a few really common ones that we use here. Cell class is just, it tells us whether the cell is excitatory or inhibitory. And that's based on any information that is available that could potentially lead to that column, which includes the morphology of the cell, any transgenic markers, if there are any. It includes whether the cell itself created an excitatory synapse onto another cell. So if you just want to divide your cells into excitatory and inhibitory, this is the way to do it. We have Cree type, it gives us access to transgenic reporters in mouse data. We've got cortical layer, and we have dendrite type, which is specifically spiny or aspiny for picking between excitatory and inhibitory cells. So these are all going to be column names that we can filter on in order to group our synapses together. But if you remember, in the database, we're querying from this pair table, and each pair consists of two cells, right? It's a presynaptic cell and a postsynaptic cell. So that means that all of these cell columns that we're looking at here appear twice in the data frame. And so to disambiguate that, we prepend their names with either pre or post, so that you can request specifically features of either the pre or postsynaptic cell. So here's an example. I'm going to ask for a swarm plot where on the x-axis, I'm looking at the presynaptic cortical layer. And on the y-axis, I'm looking at this STP induction metric. So again, this is measuring whether a synapse is facilitating or depressing. And so one of the things that pops out here immediately is that in the human data, the vast majority of our data is in layers 2 and 3. But you do see a slight difference in the distributions here, where layer 3 is preferentially more depressing, and layer 2 is preferentially a bit more facilitating. So that's a start. There's a whole lot that you can do with just what we've seen so far. But as we move forward, we're probably going to want to be able to define more complex cell categories, typically combinations of 2 or 3 different criteria. And to help with that, we have this cell class helper. And so basically what we do is we define a list of cell categories. So for example, layer 2-3 excitatory is a cell class in which the cell is excitatory and the layer is 2-3. A layer 2-3 parvalbumin is a cell class in which the Cree type is parvalbumin and the cortical layer is 2-3. So these 4 cell categories are going to be used to split up synapses in the mouse data set. So I'm going to run another query here. Again, this is standard multi-patch. I'm going to pick this time from the mouse data. I'm picking all pairs that are connected by a synapse. And again, I'm preloading information about synapses and cells both. And finally, I create a data frame from that. And we've loaded about 2,500 synapses from the mouse data. Now I'm going to use this helper function here, classify pair data frame. And the idea is we're going to take this data frame, which contains many, many synapses, and I'm going to add 2 new columns to it, pre-category and post-category. We're basically saying, take all of these synapses, categorize them based on the categories that I just defined above, these here, and the names that I gave on the left are going to become the values in these new category columns. So as a check, if I ask for all of the unique values in pre-category, I get the wrong set of names because I think I didn't run this cell. Let's just try these again. That's a little better. So layer 2-3 parvalbumin was one of the categories that I had defined here. All right, so now we can do exactly what we did before. We could just do a swarm plot or something like that, where the x-axis is a cell category instead. But I want to do something a little bit more involved. I'm going to use a pivot table. This is a really handy feature that Pandas provides. And basically what we're going to do is we're going to create a matrix where the presynaptic category is on the y-axis and the postsynaptic category is on the x-axis. And the value that I want to see is synapse latency. So for each matrix element, it's going to collect multiple values, and it's going to take the mean value and show us that. So let's see what that looks like. So as an example here, layer 2-3 SST cells have a mean latency of 1.4 milliseconds onto layer 2-3 parvalbumin cells. Seaborn has some nice tools for visualizing those kinds of results. So let's have a look at that. So this is exactly the data that we just saw, but by color instead of by number. So this whole procedure that we just did, where we run a query and we create a pivot table and then create a heat map from it, is something that we do so frequently that we just turned it into a single function called cell class matrix. And so here I'm defining 3 new cell categories, similar to what they were before, but now I'm not selecting for layer anymore. So this will aggregate cells from across all layers. And I'm going to run this cell class matrix 3 times using 3 different metrics. And the metrics names are given here. And we're back. So I've got one matrix that's comparing all of these 4 cell classes, where we've got presynaptic and postsynaptic. And this one is looking at a measure of synaptic strength. The one in the middle is looking at a measure of short-term plasticity. And the one on the right is looking at the postsynaptic current rise time, which is just a metric that describes the shape of the currents. In this center matrix, we can see some interesting things. Parvalbumin cells have very highly depressing synapses almost across the board. Excitatory synapses onto SST cells tend to be very facilitating. This is something that we've already known. And likewise, SST onto VIP and VIP onto SST are facilitating. So next I want to move on to talking about measuring connectivity. And measuring connectivity is surprisingly difficult, especially for an experiment like this. It's one thing to take a bunch of synapses and average properties. The trouble with measuring connectivity is that there are many opportunities for false negatives. And we want to be really careful about how we handle those false negatives and how we interpret the results. It's really common in the literature to see a multi-patching study like this, where you're comparing the connectivity of multiple groups, and you say, of the cells that we looked at, out of 50 connections that we checked for, we found 10. So we're going to call that 20% connectivity. If you are just interested in comparing synapse types within the context of that study, that works okay. It does have some problems, but it works okay. If you want to do anything else with those results, you run into problems. So the big thing that we're facing here is that there are cell type dependent differences in connectivity. That's what we just talked about. The next problem that we might run into is that there are spatial effects of connectivity. For example, we know that as the intersomatic distance goes up, the probability of connections between two cells goes down. And likewise, just the relative positions of two cells can have a major impact on their connectivity. So if you were, for example, trying to build a model of a cortical network, it doesn't help just to know that 20% of these connections were found in this experiment. You need to know where they were found. You need to know how far out does that 20% extend. The next problem that's going to happen is these false negatives, where we've got connections severed during tissue preparation. So any two cells that you pick may have been connected by an axon that went across the slice plane and back in. And if that axon is cut, you will not be able to detect it in the experiment anymore. For a long time, this was thought to be the major issue with measuring connectivity in slice. And in our data more recently, we found that it's not as much of an issue as it was thought to be, as long as you're mitigating those effects in the experiment. A larger issue that we run into is that many synapses have a very low signal to noise ratio when you're recording at the soma. So the somas can be pretty noisy, and the synapses themselves can be extremely small. And to add on top of that, synapses can be very far away from the soma, which means there's a lot of dendritic filtering that goes on before it gets to your recording electrode. So all of these things ideally need to be dealt with in some way. But to start us off, I'm just going to worry about this first point. I'm just going to see what is it going to take to measure connectivity when we're looking just at the proportion of cell pairs that were connected grouped by cell type. So we're going to start the same way. I'm going to run a pair query. It's going to be a standard multi-patch experiment. I'm querying from mouse data again. Previously, you saw synapse equals true, where I was looking for all of the pairs that are connected by a synapse. And now, since I'm interested in connectivity, I want to know all of the pairs that were checked for connectivity. And that's what's happening in this line. And then I'm going to preload all information about cells, since I want to group the data that's going to come back from this by cell types. And finally, I'm going to generate a data frame from that query. So to reiterate, a single row in this data frame is going to contain information about a cell pair, which represents a possible connection from one cell to another. So if we want to get the proportion of connected cells, we just need to take the number of synapses that were found in that set divided by the total number that were checked. And ideally, we're going to split the data up by cell type before we do that. So I'm going to do the same thing that I did before. Maybe I should run this query. I'm going to run classify pair data frame, which is going to add 2 new columns to my data frame, pre-category and post-category. And which cell categories are we going to actually get there? The last set of cell categories that should have been defined were here. All right, so we're going to classify the pairs in this data frame. And then I'm going to ask for another pivot table where all I'm really doing is getting the length inside where I'm looking for just the number of pairs in each matrix element. And then I'm going to do the same thing, but I'm going to count the number of connected pairs, right? So I'm going to get one table that contains the number of pairs that were probed for connectivity, and a second table that contains the number of pairs that were connected. And then I'm just going to show the quotient, the ratio of these two. All right. So in this case, excitatory to excitatory cells have an overall connectivity of just 4.6%, which is actually pretty consistent with what we've seen in most of the literature. In contrast, parvalbumin has a much higher connectivity rate across our entire experiment. So that's fairly straightforward, but remember, these numbers are maybe useful for comparing just amongst themselves, but not useful for much else beyond that. There are even biases left in here where, for example, if all of the synapses that were checked in this category were checked at very low distances relative to these, well, then we would expect this to have higher connectivity because we know that cells are more likely to be connected if they're close together. And in that case, these differences would not be due to cell type differences, but rather just due to an experimental artifact. So the next thing we want to do is try to take care of that bias. We want to actually look carefully at the profile of connectivity for each of these elements. But before that, let's just look at a larger matrix where we're actually looking at cell type and layer at the same time. And for that, I'm going to use this generate connectivity matrix, which essentially does everything that we did up here in a single function call. If I remember correctly, this one takes a while. Something else that's happening here in these data, something you can't see is how many cells, how many synapses are being probed in this. So we don't really have a good sense of the confidence intervals for these values. And when we visualize the data as a heat map, it's still, it becomes even more difficult to get a good sense of what the confidence intervals are for these. So something that we've implemented in this this function here that is still running is it will color each element by the connectivity, but also by the confidence. And the way it does that is cells that have lower confidence are going to appear more gray. And I think this will make sense when the matrix shows up. Here we are. So this is all of our mouse synapses grouped by layers, layer 2, 3, layer 4, layer 5, layer 6, and then within each layer by subclass, pyramidal, parvalbumin, SST, VIP, and so on. And you can see these gray regions here are regions that are just not heavily sampled in our data. So these are generally places to avoid if you're trying to use the data in order to seed a model. Okay, so now let's talk about modeling connectivity versus intersomatic distance. The idea here is going to be to look at the relationship between connections found versus connections probed as a function of distance between the two cell bodies. And for that, I'm using this function, show connectivity profile. And I'm going to use a Gaussian model to fit the data. I'll run this cell, and then we'll go back and talk about what's happening here. So right here, I am selecting from my data frame only synapses that are excitatory cells onto inhibitory cells. I want to look at this specific class of synapse. I'm getting a list of all of the distances between cells, between cell pairs that were probed for connectivity. And I'm getting the list of which of those were connected. And with these two things, with the distances between the cells and whether those cells were connected, I can generate everything you see here. So the connected and probed are these raster plots at the bottom. And the gray line is binned proportions of connectivity for each distance. So you can see that as we get to larger and larger intersomatic distances, the probability of connection drops down. And as we get farther out where there's no sampling, the confidence intervals, which are these gray bars, get much larger. So then I'm going to fit a Gaussian to all of these connectivity data. And when I call show connectivity profile, I'm also giving it the fit, and that is this red line here. So the output of this analysis is these two values, this P max, which is the maximum connection probability that was fit here, and a sigma value, which is the sigma for this Gaussian, about 100 microns. These two values are much more useful for designing a network model. This is exactly the kind of thing that I think you would want to start with, as opposed to the proportions connected that we saw above. I'm not going to take this analysis any further during the tutorial, but if you're interested in seeing more of this, we've got a whole lot of it in our manuscript that is up on bioRxiv right now, and there are links to that in this notebook. So the next thing that you would want to do, that I'm also not going to show you in the tutorial, but I'll just discuss briefly, is you want to correct for any bias in connectivity measurements. So we see above here that there's a steep relationship between intersomatic distance and the probability of connectivity. And so one consequence is that if two groups of synapses are sampled with different intersomatic distances, then they may appear to have different rates of connectivity, but that would just be caused by an experimental artifact. So the first thing you want to do is model that relationship, the connectivity versus distance relationship, so that you can essentially remove that bias from the equation. And the results that you get here, the P max and the sigma, are ideally very robust to how those distances were sampled in the experiment. Likewise, we have these other two major sources of experimental artifacts that I discussed earlier. You can have connections that are damaged during tissue dissection, and you can have connections that are missed due to a low signal to noise ratio. And so these artifacts are definitely present within our data set, and they're especially prominent when you compare results across studies. The reason being that any given study is going to use very different experimental protocols. So it's likely that you're going to get different thresholds on signal to noise ratio that are acceptable for any different study, and you're going to get different levels of connection damage. And likewise, you're likely to get different intersomatic distances sampled in any given study. So when you're comparing results between different studies, it's really critical to do this kind of analysis so that you're correcting for those experimental effects. So in the same way that you can correct for distance, you can also model the relationship between the likelihood of tissue damage and the connectivity. And we do that using a couple of different features, such as the distance that a cell is away from the cut surface. So this is just under the assumption that a cell that is close to the surface is much more likely to have its processes cut. We also have an anatomical measure of the length of an axon until it is truncated. And so in cases where the axon is truncated very short, you might expect not to see any connections. So we can use those properties and we can model the relationship. We can say, as these properties get worse, how does that affect connectivity? And the really cool thing is that with enough data, you can use those relationships and those models to essentially correct and remove those experimental biases, or at least produce a first order estimate of what you expect to have gotten in the absence of those biases. Same thing happens with low signal to noise ratio. And we actually find that signal to noise ratio is a much larger problem in these experiments than tissue damage. So again, I'm not going to show you any of that in this notebook, but there is a whole lot about it in our manuscript on bioRxiv. Check that out if you're interested. Drop us messages in the forum if you have any questions about it. All right, so the last thing I want to talk about is the dynamics of our synapses. And this was actually a big part of the motivation for our project. A lot of the literature that came before focuses primarily on measuring connectivity and measuring the strength of synapses. And the reason for this is really just that it's quite a bit harder to measure the dynamics of the synapses. You have to spend a lot more time probing each synapse. And we wanted to take care of that. We wanted a large data set where we were running the same standardized protocols on many, many synapses across many, many different cell types in the cortex. Unfortunately, it's actually really difficult to get the same kind of data from every synapse. And the reasons for that are pretty simple. One is just that every experiment ends when it ends. It's pretty difficult to control that. And another reason is sometimes presynaptic cells don't spike when you want them to. So in the end, after you've asked for hundreds and hundreds of spikes, you might not have gotten exactly the number you asked for. And that means that there are slight differences in the data that we collect from all of these synapses. So that, and for a few other reasons, required us to come up with a model that was capable of using all of the data that was present in the experiment, all of the spikes in whatever timing was present and the responses that we recorded, and basically come up with a simple description of the dynamics of the synapse, which really means the stochasticity of the synapse, how variable is it from event to event, and how much short-term plasticity is there. Is the synapse getting stronger or weaker when there are rapid trains of inputs? So we developed this model. Conceptually, it's pretty simple. Given a list of presynaptic spike times, what we want to do is predict the distribution of likely response amplitudes after each spike. And that's all it really needs to do. So the model has several parameters that combine quantal release with short-term plasticity. The first three parameters that I've listed here are primarily about quantal release. we have the number of release sites. And the idea here is any given synapse, any given connection between two cells is likely to have multiple sites where vesicles could be released from one cell to the other. So that is this parameter here. We have the base release probability, which is the probability that a presynaptic spike will cause any release site to release a vesicle in resting state. So that release probability gets modified as activity goes on, but if you imagine waiting a long time with no spikes, this would be the release probability for each of those release sites. Mini amplitude is the amplitude expected from a single vesicle. And then we've got a coefficient of variation on the mini amplitude, which allows that value to vary a little bit. And we've also got a measurement standard deviation, which says even if these four parameters are absolutely correct, there's still going to be noise in the recording that's going to prevent us from seeing exactly the distributions that you would expect from these parameters. And then the final four parameters are for setting the strength and time constant of depression, and the same for facilitation. So when we combine all of these together, what we have is a model that is able to replicate a pretty broad variety of different synapse types in terms of the reliability of the synapse, which is to say how much variance is there from event to event in terms of the amplitude that it's going to respond with, and in terms of the short-term plasticity. Can the synapse follow becoming weaker, becoming stronger in the same way that the data does, and does it recover in the same way? So if you induce plasticity with a sharp train and then wait, does the synapse recover with the correct rate? And this is a very simplistic synapse model, but it does a reasonably good job of capturing a lot of the dynamics of the synapses. One major issue with models like these is that once you've got all of these parameters, it's really difficult to find the best set of parameters for any given synapse. And so basically our approach to this was to brute force the problem, to make this code run fast enough that we could test a very, very large parameter space. And in doing that, it actually opened the door to some interesting analyses that are in the manuscript that I'm not going to get to talk about today. So in the end of that, though, after we've searched this entire parameter space, we come up with a set of values for these parameters that basically allow the model to best match the data for any given synapse. And these parameters are in the database, and it's possible to load those up and actually instantiate our model of stochastic vesicle release. So I'm going to show you an example here of how that works. Once again, we're going to start with a pair query. We're looking for standard multi-patch experiments from the human data, anything with a synapse. First thing I'm going to do here is create a mask. I'm looking for anything that has these maximum likelihood model parameters. There are a bunch of synapses in the data set that don't have these. And the reason is that some synapses have too little data to fit the model properly, or their signal to noise ratio is too low to fit the model properly. So those are being excluded here. And then I'm just picking a random synapse out of what's left, the 123rd synapse from this list. But you could categorize these synapses based on their cell types, and you could look at the maximum likelihood model parameters for all of the synapses within each category. That would be a reasonable way to, for example, seed a model if you wanted a network model that had synaptic variance or short-term plasticity. So the next thing I'm doing is just a little bit of code to extract a dictionary of the model parameters from this synapse. So let's run these two. And here are the model parameters for the synapse that I just loaded up. So this particular synapse seemed to run best with 3 release sites. It's got a base release probability of 76%. It's got a mini amplitude of 300 microvolts. So the next thing I'm going to do, just to demonstrate one way to use this model, is I'm going to instantiate the model with those parameters. I'm going to fabricate a list of presynaptic spike times. Okay, so I'm just creating an array with these time values, and these are all in seconds. So this is 100 milliseconds, this is 110 milliseconds, and so on. I'm going to run the model many, many times, 500 times here. And I'm going to keep track of the amplitudes that are modeled. So there are a few different ways to run the model. I could run it and just ask for the expected distribution of spike amplitudes, or I'm sorry, the expected distribution of response amplitudes following each spike. But in this case, what I'm asking for is just for it to pick a random amplitude from that distribution. So for each spike, it's going to make a guess at what a reasonable response amplitude should be. So I'm going to end up with, it looks like about 10 response amplitudes by 500 trials, right? So it's like I'm running this experiment on the same synapse 500 times. And that's all going to be collected in this array, PSP amplitudes. And then I'm just going to plot the results. So I'll end up here with a scatter plot that contains the spike times and amplitudes. And then I'm going to add on top of that the mean, a line that connects the mean amplitudes following each spike. And this is the result we get. So this is pretty similar to something you might expect to see in an experiment, where we've got 3/4 of a millivolt PSP amplitude. So that's a fairly strong synapse, but not surprisingly strong, especially given that these parameters were taken from the human data set. We see a fairly large variance in the amplitudes that you get in response to each pulse. We see synaptic depression, and we can see that it's recovering after a certain amount of time. And we even see the occasional events here that run negative, right? And this is just because of the experimental noise that I talked about earlier. So I think that's it. This is the end of everything I wanted to show you. I would love to take any questions if there are any. Potentially, I would like to hear about uses that you might have for this model, if you've got any. And if you don't want to speak up here, we would love to hear from you in our forum, which again is linked up at the top of this notebook.
Thanks, Luke. That was great. So we have a couple of questions in a Q&A. David Berling is asking, do you record the cortical area in which you found the synapse? Yes, absolutely. These are recorded first as a target region, which is to say the experimenter intended to patch a cell in V1, for example. And they're also recorded in the sense that we have the location of every cell registered to an image of the slice, which can then be registered to the Allen Institute common coordinate framework, right? So you just use the image of the slice to figure out roughly where in the atlas this cell appeared. So
that information, the metadata is there in order to generate those mappings. But so far, the cost benefit trade-off hasn't quite crossed threshold on that. So we're still waiting until there's a really strong need for that, for the data to have the individual cells mapped precisely into that atlas. But for now, it's maybe enough to say that with every cell, it came from V1 or it came from medial temporal gyrus in human, or in a few cases, other brain areas in mouse.
Great, thanks. So Domas Linkevičius is asking, is the Synapse experimental data itself available anywhere?
Yeah, so maybe I would like to hear more of what data specifically you mean, but it's very likely that the answer is yes. So in the larger databases in particular, every single stimulus and response amplitude is stored. And if I remember correctly, in the largest database, you actually get a snippet of the recording, the electrical recording of every single response. And because those recordings take up more data than these metadata parameters, those versions of the database are quite large, but you leave it downloading overnight and you've got it. So I believe anything you might want about the experimental data is available.
Thomas, we give you permission to speak up if you want. So if you want to follow up on this question, please feel free to say
No, can you hear me?
Yes.
Yes.
Yeah, so I was specifically referring to the PSPs, and it seems that they are available somewhere, right?
Yep, that's right.
Okay, thank you.
Yeah, thanks for the question. All right. So I think that's all the questions that we have at the moment. Maybe I can also add a question, Luke. Can you elaborate a little bit about the performance of the model that you described, like how it compares with some other models out there in terms of, how fast it can run, simplicity versus the computational efficiency?
That's a great question. There are a couple of other models that came before ours that are similar in their design, in terms of the way that they combine these quantal and short-term plasticity parameters. And we did try. We tried out one of those, the one that seemed most similar to ours, and we found that its performance was roughly 10 to 100 times slower, but they were also doing a lot more work, right? So there were a number of corners that we cut in order to get where we are, in order to simplify the math and the computation that went into it. So it's not to say that ours is necessarily better than theirs. It is just designed to be used in a different way. In terms of performance for, if you're asking the question, wondering whether you would want to use this in an actual network model, I would hesitate to say yes to that. I would be more inclined to just look carefully at the mathematics in the model and re-implement it, in Neuron or whatever. It's likely to be quite a bit faster that way.
So you mean in terms of the current implementation in the code, you're saying that there's probably a faster, better implementation that's possible for particular simulation software that one may want to use. But in terms of what kind of dynamics your model captures, it sounds like you're pretty happy with it and confident that it can give you a good description of the underlying reality.
Yeah, I think that's right. There are some exceptions to that. We talked about that a little bit in our manuscript. There are like some interesting things, dynamics that we saw in our synapses that the model did not capture well. But that's a trade-off, you know, it's how complex do you want the model? How worried are you about overfitting the data and so on?
Great, thanks. Yeah, thanks very much, Luke. That was wonderful. And thanks to all the attendees. So if there are no other questions, I think we are ready for a break. So let's break now. And we'll come back at the end of the hour. So 10 a.m. Pacific time or 1 p.m. Eastern time. So 17 minutes from now. Come back for the presentation by Saskia de Vries about the two-photon imaging in awake mice. We'll see you then. Thank you,
Great.
everyone. Thank you, Luke.
Thanks, everyone, for your attention.
All right, so I'm going to get started. My name is Saskia de Vries. I am an investigator at the Allen Institute who's working on our two-photon Allen Brain Observatory data set. And I'm going to do a tutorial now on that data set, what it is, and how you can access it using the Allen SDK, which is our software kit. So I'm going to start with just a brief overview of the data set. I have a few slides to kind of explain what it is, and then I will go to a Jupyter notebook, which is in our repository, and I'll get a link in the chat for you that will walk through how you access the data, how you find particular experiments, and some of the types of analyses that can be done with it. So let me get my slides up. All right. All right, so the Allen Brain Observatory data set is one of the in vivo physiology data sets that have been collected through the Mindscope program at the Allen Institute. And this program, over the last several years, we've built several data collection pipelines that allow us to systematically record in vivo physiology in the mouse visual system. And there's actually a couple of different pipelines, and you're going to hear about 3 of them over the course of today and tomorrow. So the first data set that I will be telling you about is the visual coding two-photon data set. Let me get a laser pointer. 1 second. Here we go, where we use calcium imaging to record the activity of hundreds of cells at a time in response to a battery of different visual stimuli. And we have a pipeline with many different steps where the mice kind of have a surgery to implant a cranial window that gives us optical access to the brain. We map out the visual areas using intrinsic signal imaging. We habituate the animal to the presentation of visual stimuli, and then we record the activity using two-photon calcium imaging. Once all of the data is collected from a given mouse, the mouse is perfused. We do some serial tomography that allows us to validate the expression of our transgenic line that I'll describe in a minute, as well as perform other quality controls. And then the data is organized and made available through our SDK, as well as through a web browser that we have. So our first data set that we collected from this pipeline was the Allen Brain Observatory, the visual coding two-photon data set. We have a parallel pipeline that uses Neuropixels, and Josh is going to be telling you all about this data set in the talk just after mine. But it pretty much leverages pretty much the same steps of our pipeline, except during the recording stage, he uses 6 Neuropixel probes to record the activity of thousands of neurons at a time. And so that data set was released a couple years ago, and you'll hear all about that later today. In just this past year, we've released a data set that you'll hear about tomorrow, which leverages the same type of single plane and now also multi-plane two-photon calcium imaging when the mice are performing a particular change detection task that they've been trained to detect when images change, flashed images change identity. And so that you'll hear about tomorrow with Alex Pyatt. And then a final data set that has not yet been finished collecting, and so you're not going to tell you about it, but will be coming in the near future, is the visual behavior Neuropixels data set that uses the Neuropixels recording pipeline with the change detection task. And that will be coming soon, but is not yet available. But as I mentioned, I'm going to be focusing on our 2-photon data set, which is a passive viewing experiment using 2-photon calcium imaging. And the idea of this data set was to survey the visual responses in the mouse cortex, where we could look at responses across lots of different visual areas, across lots of different types of cells in the different layers of the cortex and of different genetic properties, so different transgenically defined cells, and using a wide range of different visual stimuli that include some kind of canonical, classic grading stimuli and noise stimuli, as well as natural images and natural movies. And this data set was then designed to allow us to think about questions about how visual information is represented and transformed through the visual cortex as information moves through different areas or across different layers. How is that information being transformed? Are there differences across these cortical areas, across different cell types or different layers, and whether the stimulus statistics affect the visual encoding properties of neurons? So just to give you a feel for these experiments, as I mentioned, this is a 2-photon calcium imaging. We use transgenic lines to express a genetically encoded calcium indicator, and we use GCAMP6. And so we're expressing GCAMP6 in a particular population of neurons. I'm going to play this video, which I can't do with a laser pointer. Give me a second. Here we go. And so you can see that there's probably a few hundred cells in this field of view that light up at different times. And the mouse is on a wheel underneath the microscope. The mouse is awake and it's free to run if it chooses. And you can see different stimuli that are being shown behind it. We track the pupil, both the area of the pupil as well as its position. And then on the far right, you can see the stimulus that's being shown to the mouse, which varies during the experiment from noise and movies and gratings. And this red dot then indicates the position of the eye, the pupil position on the monitor during the experiment that we're recording. So from this experiment, you can see we can pull out lots of different pieces of data. And so this figure below shows you, for instance, we can pull the activity of different cells. We identify different regions of interest from the movie. We assign these to different cells, and we can pull out the fluorescence activity of each cell during the entire recording. We've got the stimulus presentation. So the colored bars here indicate the epochs of movies or noise or grading stimuli or spontaneous activity that are shown throughout the experiment. And as you saw in the video, we show, say 5 minutes of a movie, 5 minutes of noise stimuli, then another 5 minutes of the movie. We kind of interdigitate the different stimuli. The eye position, as well as pupil area, it's not plotted here, but we have the eye position that we pull from the eye tracking movie, as well as the running speed that gets recorded by the wheel that the mouse is on. And so all of these can be aligned, and we can then package these data into a Neurodata Without Border file format. And this is the data that we make available to the public, and I'm going to be showing you how to access it. And so each experiment, each single imaging session has a single NWB file that has all of this data, all of these activity traces, as well as information about the ROI masks, metadata about the experiment, all of the information about the stimulus, as well as the actual images that are being shown, and a lot of other kind of subtle details that go into that. So over the course of a couple of years, we used this pipeline to collect over 1,300 hours of data from over 250 mice, which generated a data set that is summarized in this table here. And what you can see, this just summarizes how much data we've collected from our different cortical areas. So just to unpack this, we've got 6 cortical brain areas. VisP is the primary visual cortex, V1, and then these are 5 higher visual areas that surround it. We have sampled both excitatory and inhibitory neurons that are indicated in this column here. And these are, either we sample them across many different layers because we're using a driver that's pan-excitatory, or we have drivers that are specifically expressed. Sometimes they're specific to a given layer. Other times they have kind of more specificity, and they label particular subtypes within a layer. So for instance, in layer 5, we've got these FESF2 and the TLX3, where FESF2 is a cortical fugal projecting and TLX3 is a cortical projecting population of neurons in layer 5. So we've got different subpopulations within our layer specificity. The vast majority of the data in our data set was collected using GCamp6 fast, but there is a small amount of data that was collected using GCamp6 slow. And so that's indicated by this column here. And so then you can just see the total numbers. This is the number of cells and then the number of experiment containers that we've collected for each of these combinations across areas and transgenic Cree lines. So again, these are the transgenic lines that we've used that label different populations of neurons, like I described a second ago. A few of them are what we call pan-excitatory, that they label pretty indiscriminately across different excitatory cells, across different layers. Others are specific to layers or to subpopulations within layers, as well as we've got these 3 inhibitory Cree lines that label our 3 large inhibitory classes, VIP, somatostatin, and parvalbumin. There's a lot more information in our documentation about these transgenic lines. But one of the things I just want to point out is that this is a point of kind of contact between the Brain Observatory data set and some of our other data sets from the Allen Institute, such as the cell types database that you'll hear about tomorrow morning, as well as the multi-patch synaptic physiology data set that you just heard about from Luke, where they also leverage many of these same Cree lines in their own experiments. And so this is a point of reference between some of these data sets. Sorry, so I mentioned there we go. We used a battery of different visual stimuli in this experiment. These include drifting grating stimuli. So these are gratings at different orientations and temporal frequencies that are moving in different directions. We've got static gratings, which are flash gratings. And again, there's different orientations and now different spatial frequencies. So the period of the grating changes for different conditions. A locally sparse noise stimulus, where we've got white and dark spots that flash in different locations in the monitor, as well as natural scenes, where we have 118 different images that are flashed, again, in the same type of presentation as we use for the noise stimulus. And then natural movies, which are a couple of different clips. These are all taken from the movie Touch of Evil. And we show these movies several times. And then the spontaneous activity, which is just mean luminance gray for 5 minutes without any changes in pattern or temporal structure. So in order to fit all of these stimuli into an experiment, we actually broke the experiment into 3 different imaging sessions. And so we would return to the same group of cells, the same field of view on 3 different days in order to present different 1-hour stimulus sets. So we have 3 different stimulus sets, session A, session B, session C, that have, say, the drifting gratings and natural movies in session A. Session B has static gratings and natural scenes. Session 3 has the locally sparse noise and more movies. There's actually 1 movie clip that gets repeated in every session. So we have that. And we also have at least 5 minutes of spontaneous activity in every session as well. So what this means is that we record from the same group of cells on 3 different days, but each session is its own separate neurodata without borders file. And so in order to kind of relate activity across different days, you have to access different files. And this is where our software kit becomes really useful for accessing these data, as well as being able to kind of work across different individual experiments or individual sessions. And so again, so we have this, the Allen SDK is our Python analysis toolkit, and we're going to be using this in a few minutes as I walk through a notebook. In order to use it, you will have to pip install Allen SDK, but it's a really great tool for accessing not just the Brain Observatory data, but actually many of our different data sets. And this allows you to, and I'll show you in greater detail, but this allows you to kind of find the different experiments based off of different metadata criteria. It allows you to access all of the different pieces of data, the activity traces, the stimulus information, as well as things like looking at the actual field of view and finding the masks of all of the different cells. So we'll dive into this in a minute. I just wanted to point you to a couple of places where you can get more information. On our website, observatory.brainmap.org, you can actually browse a lot of the data, and we have some really great dynamic visualizations of the data. But we also have a link here for our SDK, which has information on how to install it and kind of getting started with it, as well as a link here for documentation that has several white papers that explain how we did these experiments, the stimulus, real fine details about the stimulus and analysis of it, and the visualizations, and then also a deeper characterization of these transgenic lines for those of you who want to know more about them. So I just wanted to kind of, before I move on, just there's been a lot of work that's been done with this. There's 24 external publications, including preprints that I know of to date, 13 that have come out of our institute itself, that have used this data set. And so they have a lot of different types of ways of using it. Sometimes people use it in relation to their own data sets to kind of provide a little bit of a control or kind of to provide a larger context for some of their own results. Sometimes people are analyzing it and, for instance, comparing different aspects of, I think this is stimulus encode, being able to decode stimulus identity and that performance across different visual areas. Other people will take the movies, the raw movies, and I'm not going to show you how, but we do make those available. They're all on AWS, and people can have used the movies to validate and develop different methods for cell segmentation or cell matching across days. And there's a number of papers that have used the data in that way. Just to show you a couple of ways that we've used the data. So we've got a paper, a platform paper about this in Nature Neuroscience from last year. And we can, for instance, characterize different responses, visual tuning responses. So in this case, this is summarizing the direction selectivity. This is how strongly cells prefer motion in one direction compared to its opposite direction. And so this is in response to that drifting grading stimulus that we use. And we can measure a metric, we can compute a metric of that selectivity for one direction over the other. And this direction selectivity index is being summarized in these plots. This shows the median value is the color value here. For the 6 different visual areas, we've got V1 and then the 5 surrounding higher visual areas. And then across the different layers that you can see this figure is kind of repeated 4 times for the different layers that we've sampled. And then on the right, this shows a summary just within primary visual cortex across all of the different Cree lines that we sample there. So for instance, we see really high direction selectivity in V1 layer 4, which was surprising to us. We actually expected it would be higher in some of the higher visual areas than it was in V1. But layer 4 showed this very strong direction selectivity, specifically in V1. We could also look not just at single cell metrics, but at population metrics. So this is decoding performance, again, summarized across different areas and different layers and different cell types. And in some ways, this again is for decoding the grading condition, the speed and the direction of motion. In some ways, it mirrors the direction selectivity. In other ways, it's not actually a perfect match. So it suggests that direction information, there's more information than just that direction information going into that population code. We could also summarize, in this case, I'm showing a plot summarizing the coefficient of variation. One of the things we found in our data set is the variability of responses is quite high. And you can see this is true across kind of all of the different areas and cell types, although you can see this inhibitory cell, the somatostatin cells actually show less variability than most of the excitatory populations, that they actually show more reliable responses. And finally, we can look at things that aren't related to the visual stimulus. So this is a plot summarizing the correlation of activity, of neural activity with the mouse's running speed. And we see that, for instance, in V1, across all of the layers, we see a very strong correlation on average. Whereas in the higher visual areas, there's not much of a correlation, except for in layer 5, where we see a strong correlation kind of across all of the areas, but it tends to be weaker. I guess an exception here is with this area over here, LM. But the other visual areas are much more weakly correlated with running activity. So these are just a couple of examples of ways that we've started to parse the data, and there's a lot of other things that can be done. But what I want to do now is I'm going to get rid of these slides, and I'm going to switch over to the notebook so we can start diving into how to access the data and work with the data itself. So let me get that up and running. Give me 1 second. All right. So this is, hide the header just to give a little bit more space. Great. So this is a Jupyter notebook that we have made. It's available in the repository for the course, so you can access it yourself and work through it. You'll need to pip install Allen SDK in order to run it. But otherwise, we've got kind of a small number of dependencies here. So to run this, you just, for each cell, you just hit Shift Enter. This just installs some of our dependencies. This is what actually sets up our access to the Brain Observatory. So we're importing the Brain Observatory cache from the Allen SDK, and then I'm pointing it to a manifest file. So the first time that you run this on your machine locally, you're going to want to just leave that blank and just actually delete this whole part right here. And it will create a manifest for you. And this manifest is then what it uses to organize and access the data. And so once you've started accessing different NWB files, and I'll describe that in a minute, it will download a file to your local machine, wherever that manifest file is located. Or in this, if you don't give it a location, it'll go to your working directory. And then in the future times when you use it, if you point it to that same manifest file, it will always know where to find the files that you've downloaded it. So this is kind of a key thing for having everything organized and ready to go. And so you just run that. It gets, if you don't already have the manifest file, it will create one for you, and then it instantiates the Brain Observatory cache. So first, I want to introduce you to a few functions in this cache that allow you to kind of understand the data that's available, and kind of find some of the key terms that we can use in order to kind of pick experiments of key experiments of choice. And so we have a number of functions from the BOC that are, they start off as get all this and get all that. So get all targeted structures, you can see, returns all of the visual areas that we've collected data from. So VIS-P is primary visual cortex. Then these are our higher visual areas, VIS-L, VIS-AM, VIS-AL. Get all Cree lines gives you a list of all of the different transgenic Cree lines that were used. Get all reporter lines is a list of all of the actual indicators that are used. So some of these are GCamp6 fast, and there's two different versions of that, AI93 and AI148. And then a couple are the GCamp6 slow. And so I typically don't find myself using this, but there might be situations where you do want to specify. It highly correlates with the Cree lines that are being used, but there's maybe one situation where there might be ambiguity there. Get all stimuli gives you a list of all of the different stimuli that have been used. Get all session types, likewise a list of all of the session types. So we have 3 session A, 3 session B. You'll see that we have 3 session C and 3 session C2. Session C, as I mentioned before, one of the key stimuli in session C is the noise stimulus. Partway through the data collection, pretty early on, we found that the noise stimulus we had, which had 4-degree spots, wasn't very effective in the higher visual areas. And so we added 8-degree spots to that stimulus. And so in 3 session C2, it uses the locally sparse noise 4-degree and 8-degree. In 3 session C, it uses locally sparse noise, which is the 4-degree version of locally sparse noise. And so that's like one small difference in some of the data, but it functionally, I mean, you can do the same analysis with all of those different stimuli. So these are really important because if you want to go in and find data and you want to specify that you want data from parvalbumin, you need to actually have the correct string to use. And so it's useful to have these functions that help you kind of get yourself oriented so you can find the data that you want. So you can explore some of those get all functions. They kind of give you these parameters. Now we want to find some data. And so we can use some of those parameters, some of that metadata to define the types of experiments that we want. So I'm going to get the experiment containers. And I'm going to specify I want data from V1 for primary visual cortex, and using this Cree line CUX2, Cree ERT2. This is a broad excitatory Cree line that's restricted to layers 2, 3, and 4, but it's broad in its expression in those 2 layers. So I ran that cell, and this gives us, I'm making a data frame now. This gives us a list of all of the experiment containers that were collected in V1 with CUX2. And here, let me get my toolbar. Let's just add a quick cell. We can see how many experiments containers do we have. So we have 16 here. What if we just want to see all of the experiment containers in the whole data set, regardless of which visual area it is? So there's 456 experiment containers. I've whittled it down to 16 that are just in V1 and CUX2. You can see in this table, they're collected at 2 different imaging depths. I mentioned this is expressed in layer 2, 3, and in layer 4. And so we've collected data from both of those imaging depths. The donor name is an ID for the mouse. And so you'll see, for instance, here's a mouse that was imaged at a different imaging depth. So here is layer 2, 3. It was imaged in layer 2, 3. Here it was imaged in layer 4. So we will use mice to collect from different areas or different depths for a given Cree line, but you can otherwise we can't reuse them a lot for like the same layer, if that makes sense. But so you can see we've got 16 experiments for this one, and we're going to just take the first one here. I'm going to get the ID. So this is a unique identifier that identifies this experiment container. And now I want to find the sessions that are in this particular container. So I'm using get OPHYS experiments. Before I used get experiment containers. And so these are kind of some subtle term differences, but this gives us the individual sessions. And so I can make a data frame. And so you see that there are 3 sessions for the same experiment container ID. And so these are all in V1, they're all CUX2, they're all at the same imaging depth because these are the same group of cells, but they're the 3 different imaging sessions that I described, session A, session B, session C. So each session has its own, as I mentioned before, its own NWB file, it has its own ID. And we use that ID to access the data. Now you could by-step the step I used to find the different containers. You could use this metadata, the targeted structures, the Cree lines, even the session type or the stimulus type in this query for the OPHYS experiments. If you just want to just zero in on, let's find an experiment in V1 in, using ROR-B that has the natural images. And you can use those metadata to find just those sessions and pick one of them that way. All right, we already described, so the container is the set of 3 different experiments. Each experiment session has a different set of stimuli that are described in this graphic that I've also described a few times now. All right, so we've picked an experiment container ID. I want to pick the individual session from this experiment container ID that has the natural scenes. So I'm doing it here by specifying which stimulus. I could also say which session type I want. We know that that's session 3, session B, but both work just as well. So we can see I've pulled up this unique identifier for just the one session for this particular experiment in V1 with CUX2 that uses the natural scenes. And now with this session ID, I can access this data using get OPHYS experiment data. And I pass it the session ID. And this creates a data set object, which gives us access to all of the pieces of data that are within the NWB file. So we can start, I'm going to just kind of quickly show you a lot of different things that are in this file. We now are using this data set object, and we use a bunch of functions that again start with get this, get that. So get max projection gives us this image of the max projection from the movie that shows us all of the cells in our field of view. Get ROI mask array gives us an array of all of the masks that were used to pull out cells. So this object, it's an array, and the shape is 174 by 512 by 512. That means that there's 174 cells in this particular experiment, and each plane of it shows you the ROI for each cell. So just if I kind of flatten the whole thing, you can see kind of all of the ROIs in our field of view that were identified. There's a number of different fluorescence traces that are accessible in the NWB file. So let me just, sorry. So for instance, we've got get fluorescence traces, get corrected fluorescence traces, get demixed traces. All of these are different stages of we extract the fluorescence for an ROI. We then do neuropil subtraction, and you could use a function here that says get neuropil traces or get neuropil R. That gives you the correction value that was used in our neuropil subtraction for that particular cell. The corrected fluorescence traces are the traces that have had the neuropil correction performed. And then the demixed traces are when there are overlapping cells. We can demix the parts that overlap, so we can separate out the activity of this cell from that cell, even though their masks are overlapping. But the final step that we do is calculating this delta F over F. And so that's what we typically use. So here I'm going to get the delta F over F traces. It also returns timestamps that for our purposes today, we're going to largely ignore. And if we look at this is an array that's again 174 because that's the number of cells we have. And then this is the number of time points that we've collected our data on. So I'm just going to plot here the fluorescence of the first cell. So you can see here's the delta F over F. You can see there's times during the experiment where it's active, times where it's pretty silent. Here I'm just zooming in on a little window just for fun. And now I'm going to plot now the first 50 cells just to make it manageable. And you can see this is the cell that I plotted above. Here are a couple of different cells, and different cells are active at different times. So you can see kind of this cell has a big burst of epoch of activity that then shuts off, but then returns a few minutes later and then goes away again. And so you can start to think about why different cells might be active in different times during the experiment. And probably the first thing that you're going to think about is that we're showing different types of stimuli at different times. So let's find some information about the stimuli that we're using. So this function get stimulus epoch table gives us a table of the large epochs of stimulus presentation, right? So we have 10 minutes of static ratings followed by 10 minutes of natural scenes, followed by 5 minutes of spontaneous activity. Then it comes back to 10 minutes of natural scenes. And so it's not giving us each trial or each stimulus trial, but it does give us kind of the big chunks of what stimulus is being shown when. So I'm going to add this information to the plot that I made above. So the activity of 50 cells that I'm just plotting here from the delta F over F traces. And then I'm going to just create shading for each epoch of the different stimuli with a different color for the different stimulus types. And we can see now marking out the 10 minutes of static ratings, 10 minutes of natural scenes, etc. And now we can see that the different activity of the different cells starts to make a little bit more sense, where this cell that I pointed out before is really active during the natural scenes. And when it switches to spontaneous activity, it kind of shuts off, and then the natural scenes come up again and it becomes more active again. And so you can start to see some specificity in the responses that we're showing. This epoch, the pink epoch, is the natural movie that we show, and we repeat it 10 times in a row. And so you see a couple of cells here that have a very periodic response where we just see kind of this nice repeated activity at regular intervals, suggesting that there's some moment in the movie that triggers a response from that cell quite reliably across all of the presentations. All right, let's find some more of the data that we have accessible. So get running speed gives us the running speed of the mouse that I'm plotting here. And now we can add this to our plot below. And I'm just gonna, just scale it to squeeze it on here. So the Y value doesn't really make sense. We can look at the actual value here so that, high running speed is about 60 centimeters per second. So that's pretty fast running when it does run. But so the mouse kind of has a couple bursts of running early on, is pretty quiet for a while. And then we have a big long epoch of running for, a good 15, 20 minutes towards the end of the experiment. We can start thinking about how this running activity might relate to some of the activity we see from the neurons. And so we can pull up some individual cells that I find kind of interesting. So this is one cell that I really find interesting. It's actually the top row of this figure up here. One of the things you'll notice is this cell is really active during this green epoch, and that's the spontaneous activity when there's no structured pattern of the stimulus. And then you also see that it's really active in a little gap. We have 30-second gaps between some of the stimuli that we have. And so you see that, when whatever this is, the static ratings end, and then we've got 30 seconds before we start showing movies. This cell has this really strong burst of activity. And again, after this epoch of the natural scenes. And so this is one of these cells. They're kind of rare, but we find enough of them in our data set that they're not that rare. It's about, I would say, 5% of cells in the excitatory populations. It's more in some of the inhibitory populations. But we call these cells suppressed by contrast cells, that when you show patterned stimuli, their activity is actually suppressed. But when you kind of release those patterned stimuli, we actually see large increases in the cell's activity. And so this is one example of one of these cells. Here's another cell that I find interesting, which the activity of the cell, it's not perfectly correlated, but we do see that the cell seems to be pretty highly correlated with the running activity of the mouse, right? When we have this big long epoch of activity that spans different stimulus epochs, we see a lot of activity from the cell. And you see that kind of mirrored in these earlier little running bursts where we see it correlates with some activity from the cell. So this is a cell that does seem to have a strong correlation with running. This is an example of a cell that might be somewhat anti-correlated with the running, where we see more activity when the mouse is still. It's not that the cell is completely silent when the mouse is running, but it is less active when the mouse is running than when it's stationary. And so we can see already just by coarsely looking at stimulus epochs and running speed, we can see cells that show selectivity for different types of stimuli. We can see cells that show different relationships with running activity and other types of properties. All right, so moving along, let me show you a few more things. One thing I want to mention, so I've been showing you the delta F over F traces. For some of your analyses and for some of our analyses, we prefer to work with extracted events, where we've used an event extraction algorithm that was developed by some colleagues at the University of Washington to identify what we think are spiking events. These aren't individual spikes, but they're spiking events. And so this is also available. It's not in the NWB files. And so it's a slightly, instead of using the data set object, we're again using the BOC object, we're using this function called get OPHYS experiment events, and we're providing the session ID. And this returns, oh, it'll take a while, sorry, that's gonna have to download, but it returns an array that, there we go, that again has our 174 cells, the same kind of time shape. And here I'm just plotting the delta F over F in orange with the extracted events in blue for just one of the cells in our data set. And so you can see kind of where these events occur in relationship to the transients in the delta F over F. And so for some types of analyses, it's often easier to work with these event traces than to work with the delta F over F traces. So these are made available. Here I've just remade that same figure as above with the events. It's a little harder to see just because of the scale, the same types of epochs that we saw before, but it's all still there in this activity. All right, so that's kind of the activity traces that we have. Let's get some more information about the stimuli that we're showing. So I showed you the stimulus epoch table that just says this is where the natural scenes are shown, this is when the static ratings are shown. Now let's get more trial-wise information about the different stimuli. And so we can get the stimulus table again from our data set object, and then we specify which stimulus we're looking for. And again, you need to know the exact name. So if you call this natural images, it's going to give you an error. But if you call it natural scenes, it will work. If you capitalize it, it will not work. So we'll get the natural scene table that I'm plotting here. This data frame gives each row is a single trial. The frame ID is the image ID of which image is shown, and I'll show you what that means in a moment. And then it gives you the start and the end time. And these are in the imaging frames. And so this index is just directly into our activity traces that we were plotting before. Here's the table for the static grading stimulus. And so this is has a lot more information, right? We still have the start and end, and this is the frame at which a trial began and the frame at which it ended, the imaging frames. But this now also tells you the orientation of the grading, the spatial frequency, and the phase. And so for the grading stimuli, both the drifting gradings and the static grading stimuli, we have tables that give you all this information because the stimulus is generated programmatically based off of these parameters. Whereas for the natural scenes and natural movies, as well as for the locally sparse noise, it gives you an index into an array for that stimulus that says, this is the frame that is shown for this trial. This is the frame that is shown for that trial. And in order to know what those frames are, we can use the stimulus template. And so we use this get stimulus template function, and this returns an array, in this case for the natural scenes. And there's 118 different scenes, and then this is just the size of that scene that gets shown onto our monitor. And there's a warping that's been applied to it to account for the viewing distance of the mouse. So here we can look, I'm just looking at scene number 101. We can change this to scene number 103, or the corner of a park bench, if that doesn't make sense to anybody. Scene number 12 is a tiger in a body of water. So there's a lot of different scenes that were taken from a couple of different databases of natural images. And so this is one of the ones that we used. And what I want to look at now is we can take the stimulus table, and we're going to find all of the trials that we presented this image of this burrow, this hole in the ground. And I'm going to look at the responses. I'm looking for cell 1, and I'm basically just going to plot for each trial that we showed this image number 101, what's the fluorescence, the delta F over F of this cell. And so here are all of these trials. You can see the gray window is when the image is being presented. This is 1 second before and 1 second after. So you see kind of partway through a trial, we see this big increase in the delta F over F. It peaks shortly after the trial ends, and then we see this decay. But there's a lot of variability in the amplitude of this response. And then we've got other things happening where the cells, we have different images that are being shown at different times. So it could be there's a really big response to the image in this orange trial that's being shown about a second prior to this trial, and it's still, so we have this, it's still decaying from that response. It doesn't seem to respond when we show this image during that particular case. So you can see that there's a bunch of different things that can be affecting some of these responses. In fact, I just realized that what's happening is it's seeing the same image. That's this blue trace is the same as the orange trace. So that image was shown about a second apart with a couple of images in between. And that's the blue and the orange are the same trace. I just realized that. That's funny. Okay, so you can see how you can quickly just take the stimulus information, integrate it with the fluorescence information. You can start averaging these responses. You can start looking at different cells and looking at correlations. And we'll do an example of this in a few minutes. Let's see, a few more things I want to just point out from our data set that you can access. One is this dictionary of metadata. This goes beyond some of the metadata we use to identify the experiment. So for instance, we use the targeted structure and we use the genotype, the Cree line to pick the cell, the experiment that we're looking at. But there's other metadata here that could be valuable for some of your analyses. So for instance, we've got the sex of the mouse. We have, the indicator, we could have used that as well earlier. We have the session start time, which tells you kind of when the experiment was done. I know somebody who's done some analysis comparing the time of day that the experiments were carried out in to see if there were circadian effects on some of these responses. So that is one thing that could be useful. We have the age of the mouse. I've done some analyses looking at different aspects of, in this case, running activity of mice, whether younger mice or older mice run more. We don't have a huge age range. That's we try and keep things pretty narrow, but there is probably a good 30, 40 different day spread of age ranges. But you can also use the age of the mouse to, if you're looking across the 3 sessions for the same group of cells, you can know the order in which they're taken by looking at the age in days. We have also here the device name. This is the specific microscope that was used to collect the data. So we have 5 different microscopes. Most of the data was collected on 2 of them, but we have used 5 microscopes to collect this entire data set. And so I have a colleague who did some analysis comparing different aspects of again, I think running behavior, but whether mice on different microscopes ran more or less than others. And it turns out one of the microscopes, they ran a lot less. So that's an interesting little tidbit. But so there's this metadata that might be useful for identifying or for analyses. Another thing that's important to kind of point out is that we have cell IDs for each cell that we record. So I showed you there's an experiment container ID, there's an experiment session ID. These are unique identifiers for those containers and sessions. We also have unique identifiers for each cell. You can get this for a data set using the get cell specimen IDs function, which returns an array of all of the cell IDs. And so this is ordered the same way that the fluorescence traces are ordered. And so what this means is that within an experiment, the cell has an index. So if you want cell 1 of the fluorescence traces, if you want to use that index 1, but you want to know what that cell ID is, you need to then use this function to map between the cell IDs and then the index within the local experiment. And where this becomes important is that if you want to compare cell across different imaging sessions, it's going to have a different ID within each session, because we might not find the same group of 205 cells in session B as we find in session A. And so what you think is cell 1 in session A might be cell 3 in session B, or we might not even find it in session B. That's also possible. And so, but you can use the cell ID in order to say, I have a cell. We can pick one of these IDs here. I just picked one from this. It has a local index of 110, but it is cell ID, and I can go to session B and say, what is the index for this particular cell ID? And then I can map between the two. And so that's a really valuable thing that you kind of need to know about in order to do that cross-session matching. All right, so I'm now going to show just a nice little example, nothing super complicated, but we're going to look at signal correlations and compare them for pairs of cells as a function of distance between the neurons. And so this is, I'm going to do this just for the data set that we're working with right now. So I'm going to look at the natural scene responses. And the first thing I'm going to do is compute the mean response of each cell to all of the images that we show. So there's 118 different images. And we have 174 different cells. We've seen this already looking at the fluorescence traces and looking at the stimulus template. And now what I do is I'm just creating an array for the number of cells and the number of images, and then I'm iterating through all the cells, all of the images. I'm pulling up the trials of the image, and then for each cell, I'm taking the average of the activity. I've shifted, I'm just going to scroll up to show you from our example. If you remember this response, it peaks kind of after the given trial, right? And so I'm using a window of 15 frames, so that's probably about half a second starting at the onset of this trial. So basically, yeah, half a second. The first half of that is when this particular image is shown, and then we're including some of the decay in what we're averaging here. So I can run this, and this gives me an array of all of the responses. That'll take a minute to run because there's a lot of cells, there's a lot of images. But we can start to then see kind of what this looks like. So here I've already plotted it for one of the cells where you can see that there's a couple of images. Actually, what is this, like six images that have large responses, maybe another 4 or 5 that have moderate responses. And then most of the images it doesn't have a strong response to. Down here at the bottom, this is actually a pretty broad response. Most of our neurons are really selective for 1 or 2 images in our image set. Sorry, this is still running and we're not going to be able to, there we go. Okay, so we can replot that. We can look, here's a different cell. This is the cell that I showed the plot above of all of the trials where it really responds strongly to image 101 that we showed before. It maybe has some intermediate responses to 3 or 4, but it's really selective for that one. Let me just pull up some other random cells. You can see they're usually fairly sparse in their response. This one's pretty broad, but none of these responses are very large if you look at that Y scale. So that would, I think, is more just kind of noise. But all right, so we can look at a bunch of different cells here. Now what I'm going to do is look at the signal correlation between all of the neuron pairs. And so this is taking these the mean response across all of the images and seeing how correlated that is across pairs, all of the pairs of cells. So I'm just making an array of the number of cells by the number of cells, and then computing the Pearson correlation between each pair. And that will also take a second, but you can see here's that array. So, the diagonal, of course, is 1. We really only, this is just duplicated, but we can just focus on the upper portion here and see some cells are more correlated than others. So now we want to look at the distance between the neurons. And if you remember, we had that ROI mask. Let me scroll up for you. I know scrolling, it can be kind of annoying when you're watching these things, but here are the ROI masks for all of our cells, and we can find their location in this field of view and just calculate the distance between each mask from the others. And so coming back down, So here's what I'm doing. I'm finding the location for each mask. So basically where that ROI is located, I'm finding the mean of the ROI in X and in Y, which gives me the center of the ROI mask. And then I'm calculating the distance between that location for each pair of cells. So this is just Euclidean distance, X and Y, between each pair ij of the number of cells. And that gives us this array. Again, the distance is 0 along unity, but up here we can see the actual distance, and some cells are closer together and others are further apart. And so now I want to look at how the signal correlation relates to the distance between the pairs of cells. And so I'm just going to plot the distance as the signal correlation as a function of distance. Because this array is mirror image, I'm only going to use the indices for that the upper triangle, and that this function in NumPy gives us the indices just for the upper triangle, and then we can just limit it to that. And so it wouldn't look much different if I included everything. It would just be duplicated data. But and this is the cloud that we see. And so here, yeah, we've got the signal correlation as a function of distance. You can see, our very highest signal correlation is at a very close distance. And so that maybe it makes sense. Most likely this might be an overlapping ROI that didn't get demixed properly. That's possible that is in the data set. We also have, one of our lowest signal correlations is something right on top of each other too. So that's a little strange. For the most part, this looks fairly flat, but I do think that there are more higher values at closer distances than at longer distances. So there might be some structure here, some relationship between distance and the signal correlation, but it seems to be fairly weak, at least for these layer 2-3 excitatory neurons that we're looking at right here. All right, so with that, I want to just wrap up. The point of this was to just give you a flavor for the types of analyses you could start thinking about with this data set, and to point you to the tools that allow you to access these different pieces of data that are available in our NWB file. And there's clearly a lot of different things that you can do with this, and a lot of ways to kind of extend it in different directions and relate it to some of the other data sets that the Institute has generated. So I'm happy to take any questions. Let me see if I can find the Q&A window in case there are any there. I'm going to stop sharing. Here we go. So yeah, let me know if there are any questions. We have a few minutes before the next talk begins. So I'm happy to kind of hang out and answer any questions people might have. And I realized that link didn't get put up earlier, but it's now in the chat. I apologize for that, but apparently somebody else provided it. So I'm going to turn this off and put our schedule back on. But if anybody wants to talk about anything, just let me know. But otherwise, I think Josh will start in about 9 minutes from now at the top of the hour. All right, thanks. Hi, Devor. Yeah, thanks for that question. We are recording this, so I think we will make this available. I'm not quite sure how, but we will try and make this available. But yeah, that should help you go through the notebook. I also wanted to point out, I forgot to mention this earlier. So if in the repository where the notebook is linked, where the notebook is located, we also have a cheat sheet, which is a PDF. I have a printout of it next to me that kind of summarizes a lot of the key details about the experiment that I discussed, as well as some of the key functions from the SDK. And so if you're getting started using the data set, that's also a really useful resource as you start wrestling with the SDK. So I just want to mention that as well. But yeah, we will. I will see what we can do about making the video of these sessions available as well. So thanks for that. All right, a more sciencey question from Devor, so thank you for that. He asks, why was it chosen to record both visual and motor activity at the same time? Is it too stressful for the mice to not be allowed to move, perhaps? So yeah, that's a great question. So the mice are head fixed under the microscope, which we need to do in order to have optical access. But they are awake and they're on this running wheel. And the reason we did that is because kind of as we were developing these experiments, there had been some recent literature from Chris Neal's lab is one lab, and then a couple other groups had shown that when mice are running, there's an enhanced kind of arousal state and enhanced visual responses. And so the awake but stationary versus the awake but running are kind of different arousal states in the mouse. And so we wanted to try and either push the mouse more towards the more engaged arousal state, or at least be able to look at that as a function of how it's affecting the responses, because that quiet wakefulness is actually kind of almost a dulled state. And so we wanted to kind of be able to look at that arousal level in this. And so that's why we allow them to run. The pupil area is another metric of arousal. And so you can use the eye tracking data to get the pupil area and look at that, how if different types of responses change with the based off of the pupil area and the arousal state of the mouse. So that was kind of one of the motivating scientific questions that went into the experiment design. All right, I will turn it over now to Josh, who will we will tell you all about the Neuropixels data set. And thanks everybody for your attention and for participating. So thank you.
All right, thanks a lot, Saskia. So yeah, I'll be talking about the visual coding Neuropixels dataset, which is highly complementary to the 2-photon imaging dataset that Saskia just talked about. I'll start out by giving a presentation that gives an overview of what's included in the dataset and some of the scientific results that we found so far. And then I will transition to a tutorial to show how to access the data via the Allen SDK and perform some basic analyses. So I will start out by posting a link to where the tutorial lives on GitHub. So if you want to follow along while I am going through the tutorial notebook, you can either bring up the notebook within GitHub just as a preview, or you can download it. And if you have a Python environment with the Allen SDK installed, you can run the various cells in real time. I will say that the NWB files that contain the data are quite large, and so once you try to access the data for the first time, it needs to download that file to your local machine. The files are in the range of 2 gigabytes, so depending on your connection speed, it can take 20 to 15 minutes to actually download the whole thing. So if you do want to follow along, then I would recommend getting started with running the notebook now, and then hopefully some of the data will be downloaded by the time I start going through it. But that's totally optional. It's also fine to just listen along as I'm presenting the notebook and then go through and run it at your own speed later on. So let's get started with the presentation. If you have questions, feel free to drop them in the Q&A. I'll probably take a little break to answer some questions at the end of the presentation before the tutorial, and then again at the end of the tutorial. So yeah, as I said, I'll be talking about our Neuropixels visual coding dataset, and keyword in the title is Neuropixels. So if you're not familiar with what these are a new type of silicon probe that was co-developed by the Allen Institute along with scientists at HHMI Geneva and University College London in very close collaboration with engineers at IMEC in Belgium. And so the rationale for creating these probes was we wanted to shrink down the hardware required for acquiring, amplifying and digitizing neural data into a very tiny chip that was actually connected to the shank containing the electrodes. And so this allows us to pack up to 1,000 electrodes onto this 10mm shank. And then the rest of the data acquisition system is very, very compact. So this allows us to not only record from more channels per area than was previously possible, it also allows us to insert multiple probes into the brain simultaneously because the package is so small. In our case, we can stick 6 probes into the brain at once and record from up to a dozen different brain regions at once. So this gives us a really rich ability to acquire spiking activity from the brain, which is highly complementary to the 2-photon calcium fluorescence data that is collected in the imaging experiments. So I just want to give a quick plug for the Allen Institute Neuropixels workshop, which is happening in September. If you're interested in performing experiments with Neuropixels probes, whether or not you've ordered the probes already, I highly recommend applying for this workshop. It's going to be virtual this year, so no travel required. It's going to be a 2-day workshop where we teach you the basics of how to set up Neuropixels experiments, how to spike sort Neuropixels data, and kind of all the tips and tricks that we've acquired through the process of setting up these experiments at a large scale. So yeah, check out the link at the bottom if you're interested in signing up for this. So yeah, so the Allen Brain Observatory encompasses several different pipelines that allow us to collect in vivo physiology at a large scale. And there are both pipelines that use 2-photon imaging to monitor the activity of cells in the cortex, but we also have pipelines based on Neuropixels probes, which allows us to also measure activity in subcortical regions in addition to cortex. And one of the great things about the way we've set up these pipelines is that all the steps are virtually identical up to the point of the neural data acquisition. So we basically have mice that go through the same set of surgical procedures, retina-on-topic mapping to map out the various visual areas, and habituation prior to the point at which they are put into the experimental apparatus. And then we record the activity of cells in their brain as they're exposed to various visual stimuli, or in some cases perform a visually guided task. And so this makes it much easier to compare data across experiments, both within the same modality, so comparing across experiments that use electrophysiology, but also between modalities. And we have a preprint that came out last year where we explicitly look at this, trying to compare 2-photon imaging and electrophysiology data, and seeing where discrepancies arise and how we can account for those. But that's beyond the scope of what I'm going to talk about today. Here, I just want to give an overview of what is involved in our Neuropixels electrophysiology experiments to give you some more context for understanding the data that can be retrieved through the Allen SDK. So this is what our Neuropixels rig looks like. So in terms of the running wheel that the mouse is standing on, as well as the visual stimulus monitor, which is you can see on to the off to the right. Everything about this rig is the same as in the 2-photon imaging experiments, except instead of a 2-photon microscope, we have 6 Neuropixels probes that are positioned above the mouse's head. And there's a lot going on here, so I'll show you kind of a pared down image where you can see the mouse on the wheel, and then each probe is mounted on an independently movable 3-axis manipulator. And so we can then use the retinotopic maps shown in the middle image here of the mouse cortical visual areas, and then target our probes precisely to each of these areas. And one of the big advantages of these Neuropixels experiments, at least compared to the single plane 2-photon imaging experiments that Saskia talked about, is that we can target multiple cortical areas simultaneously. And so in any given experiment, we record from up to 6 cortical visual areas. You can see the little white dots indicating where each probe is inserted into the brain. And then these probes extend down through all layers of cortex and also into the subcortical areas that are below it, which include hippocampus and thalamus. So we acquired data with the OpenEphys GUI, which is software that I've helped develop. We have a grant through the NIH that funds the integration of OpenEphys and Neuropixels probes. So if you're interested in acquiring your own Neuropixels data, I highly recommend checking this out. After each experiment, we clear the brains using the iDisco method, and then look for the probe tracks, which are labeled with a fluorescent dye. And then we have a manual registration procedure that allows us to trace the probe tracks and click on key points in each of these brains, and then register all of our recording sites to the mouse common coordinate framework. And this is super helpful because it makes it much easier to compare data across experiments when you know exactly where in 3D space each of your cells was located. So basically for each individual brain, we warp it to the reference brain, the Allen mouse common coordinate framework reference brain, and then determine where each of the cells that we recorded from is located in that reference space. And so this shows on the left, you can see how we've color coded each of the different areas. And on the right, you can see aggregate across many experiments where the cells that we recorded were located. This is just looking from kind of the back of the brain to the front of the brain, projecting all the cells onto a single plane. And yeah, you can see that we were sampling densely from cortical areas as well as hippocampus and thalamus in this kind of more posterior region of the brain. So this is an example of the type of data that we can collect. So these are the cells located across 8 different visual areas, 6 of which are in cortex. So V1, LM, RL, AL, PM, and AM are all cortical visual areas. And then subcortically, we have LGN and LP, which are visual thalamic nuclei. We also have a ton of data from hippocampus, most of which is not shown. You see the green trace labeled HPC, which is the local field potential recording from hippocampus. You can see a very prominent theta oscillation because the mouse is running during this interval. And yeah, we also track the running speed as well as pupil diameter. And so yeah, the thing to note about this is how incredibly rich these data sets are. To be able to record single unit activity across a diverse set of visual areas all simultaneously. So you can look at not only characterize the properties of individual cells, but also look at interactions between different areas, measuring how information flows between different parts of the visual system on a trial by trial basis. These data sets give us access to extremely rich information about what the brain is doing. So the visual stimuli that we used are very similar to the ones that were used in the 2-photon imaging brain observatory. So we have a stimulus set that we call Brain Observatory 1.1, which is basically a concatenation of 2 of the sessions from the 2-photon imaging experiment. So one of the one of the disadvantages of electrophysiology is that at least with the acute experiments that we're doing, we can't return to record from the same cells day after day. We basically get one shot to record from a certain set of cells. And so if we want to characterize their responses to a broad range of stimuli, then we need to show all those stimuli in the same session. So in this one session, we use a flashed Gabors, just masked drifting gratings to figure out where in visual space the cell is responding to. So that gives us receptive field information. We also show brief flash stimuli, which gives us information about the onset of the response to a visual stimulus across different areas, because electrophysiology has very precise temporal information down to a sub-millisecond level. And so these brief flash stimuli allows us to monitor how activity propagates on a millisecond to millisecond basis across the visual system. And so then we also have drifting gratings, natural movies, static gratings, and natural images in the exact same parameters that were used in the 2-photon imaging experiments. And so this whole stimulus lasts about 150 minutes or so, and then there's at the end we perform an optotagging procedure. Some of the mice express channel rhodopsin in a subset of cortical interneurons, and so we can use optotagging to figure out which cells express the gene of interest. So we have a subset of experiments that are performed in parvalbumin positive, somatostatin positive, and VAP positive mice, or so the channel rhodopsin is inserted into those cells, and then that gives us the ability to tag those cells through optogenic stimulus. And of course, we also record from all the other cells in nearby, but just kind of at the end, it gives us this extra layer of information about the likely cell types of some of the cells that we recorded. So then we have another stimulus that we call functional connectivity, which uses, again, similar stimuli as we're using the 2-photon imaging experiments, but shows them with more repeats. And this high number of repeats allow us to get enough spikes to measure functional interactions between different visual areas. And in addition to the Gabors, flashes, drifting gratings, and natural movies, we also show some dot motion stimuli, which allows us to see the responses to cells to dot motion fields moving at different speeds. So this is an example of what the data for one cell looks like once we've aligned it to a visual stimulus. This is just a simple light and versus dark flash for a cell in primary visual cortex. 75 trials of each. You can see very precise temporal response to the different flashes, and there's a different delay to the light flash versus the dark flash for the same cell. And this is the same cell showing its responses to different drifting gratings. So drifting gratings are shown 15 trials each per combination of temporal frequency and direction. And you can see that this cell is highly direction selective and also has a preference for lower frequency stimuli. But you can also see, interestingly, in the star plot on the right, that it's has a direction reversal in its direction preference as you go to faster, or as you go to higher temporal frequencies. And then again, same cell, but now showing its response to receptive field mapping. So here we have a stimulus that's shown at 81 different locations on the screen, all randomly interleaved. And then for any one of those locations, we can create a spike raster in response to 45 different trials, and then create a PSDH, and then sum over these PSDHs to create this kind of heat map of the cell's response, which we call its receptive field. So this gives you a sense of where in visual space the cell is preferentially responding. And if we do this across all the cortical cells in the data set, you can see these gradients of receptive field preference as you move across areas, and you see also these reversals in the gradients. And so this nicely matches what we see when we do intrinsic signal imaging or wide field imaging to map the preference of the entire cortical surface. But now we can see this by aggregating across individual cells that were recorded throughout many experiments. You can also see this in subcortical areas. So there's a clear gradient of elevation preference in the LGN. It's a bit less apparent in LP, but on average, you also see these gradients of elevation and azimuth selectivity. So we have metrics such as receptive field location, firing rate, and a variety of other functional metrics that are pre-computed for every cell. And so these get computed and packaged as part of our NWE files, and that's how we deliver the data. The result is that there are already a wide array of pre-computed metrics for the cells in the data set, which will allow you to kind of dive into the analysis without necessarily having to compute all of your metrics. But these should allow you to look for potentially interesting patterns in the data and then dive in deeper and do your own analysis. And on the right, you can see the total number of cells that are included in our data release, sorted by the area that they're located in. So yeah, you can see lots of cells from cortex, hippocampus, thalamus, some from the midbrain as well. One thing to note here is that although we include the data for every cell that we record, there are variations in the overall quality of the recording for each cell. So you may have heard things like single unit versus multi-unit recordings. What we found is that there's not a clear distinction between single unit and multi-unit, and really there's more of a gradient in quality. So we include a wide range of quality metrics for each cell, and then by default we only show you the cells that we think are of reasonably high quality. So those are the numbers shown not in parentheses, and then in parentheses is the total number of cells that were recorded per area. And so yeah, we filter the cells by default based on a variety of quality metrics, such as presence ratio, ISI violations, amplitude cutoff. There are a lot more documentation about these different quality metrics on the Allen SDK read the docs site. But yeah, this is something to note, is that by default, when you access the data, you're only shown the cells that pass our default quality metric thresholds. But if you want to access all of the cells in the data set, you can disable those filters when you load the data. So before I jump into the tutorial, I just want to talk very briefly about kind of the first major scientific findings from this data set. So these were published in Nature, came out online earlier this year, and came out in print, I think, in April. So yeah, this is the title of the paper: was survey of spiking activity in mouse visual system reveals a functional hierarchy. And in this study, we compared our functional data, the spiking activity that we recorded across all these visual areas, to anatomical data from the mouse connectivity atlas. And so there was another paper in Nature that came out in 2019 from the Allen Institute, where they used data from hundreds of injections of viral tracers into various cortical and thalamic areas, and looked at the reciprocal connectivity between these areas, and used this information to create a hierarchy score for every area, and basically was able to map the hierarchical position of every one of these areas in the mouse brain. And then you can zoom in on just the visual areas that we recorded, and that gives you kind of the hierarchical position of these areas. And it's important to note that there's a ton of feed-forward and feedback connections between every set of areas. So it's not kind of like a strict step-like hierarchy. It's more of a shallow hierarchy, hierarchical gradient across areas, but the hierarchical differences between these areas is highly significant. And so it does make sense to think about these areas as forming an anatomical hierarchy. And so when we looked at our functional metrics, such as receptive field size, which has been shown to increase along the hierarchy in primates, we see a very strong correlation between the anatomical hierarchy score, which is obtained entirely from these tracing experiments, and the receptive field area, which comes entirely from our functional experiments. And we see a similar trend for other metrics, such as time to first spike. So as you go higher in the hierarchy, it takes longer for the information to propagate to those areas when you flash a stimulus in the visual field. And so found a very not only strong evidence for hierarchical processing in the mouse brain, but a surprisingly strong correlation between this metric obtained from the connectivity data set and a variety of metrics that we obtained from our Neuropixels data set. So encourage you to check out that paper in order to learn more. I also encourage you to check out many of the publications that have come out using this publicly available data set. So already, I think there have been about a dozen, I think there are even a few more now, publications from groups outside of the Allen Institute that have accessed our data and used it to perform their own analyses. We see kind of three general use cases for the data. So one is where basically people download the data set and use that to generate new insights about the brain, and rely almost exclusively on this Neuropixels data set. So example of that is the Fritschia et al. From 2021 by Archive, where they showed that there was the very strong dependence on stimulus history that lasted for many trials in the response to gratings. And this was primarily seen in the cortex and not in thalamus. Another use case is where people use this data set to benchmark new methods, or such as the paper from Peterson et al. In Bujaki lab. And then it's also common for people to take our data set and use it to compare to data that they've also collected. So the paper from Stringer et al. Compared our Allen Institute Neuropixels data set to some 2-photon imaging data that their group had collected. So yeah, there's a wide variety of use cases, and yeah, if you need some inspiration for how to use this data set, there are a number of publications that already use it to do novel science. So yeah, I'll just point out before I jump into the tutorial how to access the data. So the easiest way to get the data is through the Allen SDK, which does require a Python installation. But once you have your Python environment set up, you can just pip install an Allen SDK and then use the functions in the Allen SDK to download the data. Data is also available as NWB files from the Dandy archive, as well as a AWS public data set. There's more information about this on the Allen SDK read the doc site. So yeah, if you get stuck or just want to find out more, there's a ton of additional tutorials and documentation on allensdk.readthedocs.io. But if there's something that you can't find there, then feel free to post make a post on the Allen Brain Map community forum. We will be more than happy to get back to you there. Okay, so that is that is the end of my presentation, and now I will jump over to the tutorial. All right, so this is the tutorial that I'm going to be running through today, the link to which I posted earlier in the chat. Maybe I will just post that one more time in case people are just joining. Okay, so yeah, the tutorial is divided up into 5 parts. May not get to all of them in the next 30 minutes, but if anything that we don't get to, you can follow along on your own later on. So first I'll be talking about how to access the data, LNSDK functions that are used to download the data and kind of query what's available. Then I'll show how to read data for an individual session from an NWB file. Then show how to align the data to stimulus presentations, which is one of the most common things that you're going to want to do with this data set. Then I'll show an example of how to obtain receptive field maps for a set of cells in visual cortex. And then if there's time, I'll show a quick example of how to decode natural images purely from neural activity. So if you want to follow along yourself, not just read through the preview of the tutorial on GitHub, but I'll actually run the cells, recommend creating a fresh Conda environment. So this requires that you've installed Anaconda or Miniconda on your machine. But then once you've done that, you can follow these steps to create a new environment and install the necessary dependencies, and then launch a Jupyter notebook that will allow you to run each of these cells. So data access, there's a ton of data available. You can download data for individual sessions, or you can download all data for all of the sessions. There's also data is available, as I said, through the Dandy archive and AWS public data set. Yeah, more information. If you just want to download the NWB files individually, you can do that through the Dandy archive. Or if you know how to set up a notebook on AWS, you can actually download, you can run notebooks such as this without having to download the data because it's already stored on Amazon Web Services. But today I'm going to talk about how to use the Allen SDK to retrieve the data. So this is kind of our recommended method for accessing the data, because not only does it make it easy to keep track of which files you've downloaded, but it also gives you access to a bunch of helpful functions for loading the data and aligning it to stimuli. So there's a bunch of imports that are required. So the Allen SDK relies heavily on the NumPy, Pandas, and Matplotlib libraries. And then we have some custom functions that we need to import. The most important one of these is the ECFIS project cache, which is the object that keeps track of what data you've downloaded, and if data is not yet downloaded, it will grab it for you. So the first step is to figure out where you want to save your data. So you basically need a local directory where you can store your data. If you want to download everything, all 58 experiments, it's going to take up 855 gigabytes of space. So make sure that you have plenty of room available. But for this tutorial, it only downloads one large file. So I think it's in the range of about 2 gigabytes. So I'm going to start by defining our data directory and setting the path to this manifest.json file. And that's what's going to keep track of all the data that's been downloaded. If you delete this file, then it will create a new one and try to download the data again. So be sure to keep track of this file. So first, we're going to instantiate our cache by creating ECFS project cache that points to our manifest file. And then there are 4 main files that are downloaded via this cache that provide an overview of what's in the data set. So there's sessions, probes, channels, and units. And each one contains basically information related to its name. So sessions will give you all the information about the 58 sessions that are available. Probes gives you the information about all of the probes that were used across experiments. Channels has information about the specific channels within each probe. And this is helpful because it contains the CCF location, the registered location in the common coordinate framework for each of these channels. And then units contains metadata about all of the units that are recorded across all of these sessions. So we're going to start out with sessions. So the function here is cache.get session table. And this will load in our session object. This will load in the sessions table from session.csv. And then this gives us information about all the sessions, so including the specimen ID, so the ID of the mouse that was recorded. Session type is the stimulus set, either brain observatory or functional connectivity. The age, sex, genotype of the mouse, as well as some information about the recording itself, so the total number of units that were recorded, the total number of probes that were used, as well as the structure acronyms of all the areas that were recorded. So if you're only interested in experiments that recorded from LGN, for example, you can basically filter this table based on all the rows that contain LGD, dorsal lateral geniculate, in this column. So just an example of how to filter this data frame. Here, it really helps to understand how to use the pandas library. A lot of the functions in the Allen SDK rely on pandas and return pandas data frames as the main analysis object. And so here, if you're not familiar with pandas, I highly recommend reading through that documentation, because that will help you learn how to interact with these data frames. And once you know how to perform filtering operations on pandas data frames, it will make you much more productive. So here's an example of taking this sessions table and filtering for male mice that are somatostatin Cree crossed with channel rhodopsin reporter line, were exposed to the Brain Observatory 1.1 stimulus and had a recording from VisL, which is LM in visual cortex. And so we can perform this filtering, and that returns 3 sessions that match these criteria. So if you have particular type of mouse that you're looking for, or a particular area that you want to analyze, you can filter down this table to figure out the sessions that meet your criteria. We can also get the probes table. So the 332 probes were used across all these experiments. These are not necessarily unique devices that were implanted, but basically, like, within each session, up to 6 probes are inserted. And so this is the total number of unique insertions that occurred. And so for each probe, you can get things like the sampling rate, the name of the probe, whether or not it has local field potential data available, as well as the total number of units that were recorded by each probe. And then you can also look at the channels, as well as the units. So units table is particularly helpful because it includes many of the metrics for these units. And so you can kind of filter do some meta-analysis across many experiments, as well as kind of honing in on which experiments may be most useful. For example, if you want to find units with a high degree of receptive field overlap, you can search for those within the units table and then figure out which experiments those units came from. So as I mentioned before, by default, the analysis decay only shows you units with high quality based on a variety of quality metrics that we compute after spike sorting is performed. If you want to disable these filters, which are based on ISI violations, amplitude cutoff, and presence ratio, you can add some additional arguments to this get units function, and then it will return more units. As you can see, about 40% of units, so 40% of around 100,000 units are past the default quality metric. So you do get a lot more units if you disable these filters. However, the quality of some of these units is questionable. They may drop out of the recording, so they're not recorded for the entire session, or they may have a high degree of contamination, meaning there are spikes from nearby cells that are leaking into this spike train. So depending on what analysis you do, you may want to be a bit more stringent or more lenient with where the quality metric thresholds that you use. So a last thing I'll show for data access is just how to make a plot of the location of all these cells in the common coordinate framework. So this is basically accessing the units table and figuring out the CCF coordinate for all the units that were recorded, and then plotting those in 2D plane, and just adding some labels for approximate location of cortex, hippocampus, and thalamus. So this is aggregated across 58 different experiments, and the location of all the cells in this 3D space. All right, so that's how to access the overview of the data set, but presumably there's analysis that you want to do that requires access to individual spike times or local field potential traces. So in order to do that, you need to download NWB files and access data from the NWB file. So we're going to pick one of our the session IDs from our filtered sessions table. Filtered session.index.values2. So the second, actually third, because it's zero-based indexing in Python. The third session that we filtered out based on our criteria, and then we will request the units table for this session. So this will return just the units that were recorded in this one session. This one takes a little while to read out the units from the file. So this will give us a table that's kind of a subset of the larger units table that we were looking at before, for just the units that are located in this session. And then as a next step, we will filter this table. So this table is, again, just a pandas data frame. So we can filter it for units that are located in VisP, primary visual cortex, and which have ISI violations below 0.1. So yeah, you can see this table just loaded. It's got 89 columns, so lots of information about each unit. And then now we'll filter this table to get 35 units from V1 that have a low level of contamination as measured by this ISI violations metric. And so next we will choose one unit, just choose the first unit in this table, and read out the spike times from this file. So this will give you the time in seconds of all the spikes that were measured from this one unit. And you can see this warning here that says session includes invalid time intervals. And so this is something that affects a small fraction of the data sets, but it's important to keep in mind is that sometimes something went wrong with the recording on one probe. The data dropped out for one probe, or sometimes there was a problem in the stimulus display. And rather than throw out the entire data set, we just marked when these invalid intervals occurred. And so if this warning pops up for a session, you can just type session.invalid times, and it will say the interval in which the data has been affected and kind of what went wrong. In this case, probe E dropped out for a short period of time, about 20 seconds. And so in this case, this unit comes from probe C, so the spikes for this unit would not be affected. And so this will, you can also read in the waveforms for individual units. This may actually take too long, so I'm going to stop it. But yeah, you can get the data for each unit's waveform, both its peak channel as well as the data, the average waveform across the entire probe. You can also access the stimulus names for each individual session. So in addition to the neural data, you're going to want to know exactly what stimuli were presented and when. And so you can access this session's stimulus table. This also takes a little while to do. So I'm going to skip that and just get the stimulus table for the drifting grading stimulus. So session.get stimulus table for a given stimulus name will return the stimulus table for that particular stimulus type. And so that includes information about the start and stop time of each presentation of that particular stimulus, as well as the parameters that were shown within that presentation. So for drifting gratings, it would give you the direction as well as the temporal frequency of the grading that was shown. I'm not sure why this is taking so long, but I'll move on to the behavioral data. So for the behavioral data, you can get information about the mouse's running speed. Just session.running speed will give you the mouse's velocity in centimeters per second throughout the whole trial, or throughout the whole experiment, as well as screen gaze data. So if you do session.get screen gaze data, it will show you for each point in the experiment mouse's pupil area as well as where on the screen the mouse was looking. And so in this example, we can just plot it for the whole experiment. And you can see that pupil area stays small throughout most of the experiment, but there are some fluctuations in pupil size. And then at the end, the pupil gets really big. And the reason for that is because during the final presentation, during the optotagging session, portion of the session, the stimulus turns off and the mouse is in darkness. And so the pupil gets really big. Let's see, do we have a stimulus table yet? No. All right, I'll just keep going. So the third part of this tutorial is focused on how data gets aligned to stimulus presentation. So many experiments that you want to do will look at how neurons respond to particular types of stimuli, looking at either raster plots or peristimulus time histograms, or metrics that are computed based on responses to different types of stimuli. And so to do this, we can take advantage of some of the functions within the Allen SDK that are optimized for reading out spike times and aligning them to particular stimuli. So the presentation table is the pandas data frame that contains information about all of the stimuli that were shown And this shows you another way to read out the data for a particular stimulus type. You can just find the stimulus presentations in which the stimulus name matches one of the stimulus names. And then that gives us start times for all presentations, as well as the ID numbers for all the presentations, such as the index value of those rows in the table. And then we can feed that into a function called presentation-wise spike times, where we take our units of interest, which were these units from primary visual cortex, as well as the stimulus presentation IDs, and then that returns another pandas data frame that contains the spike times relative to the start of each of these presentations. So you can look at this column time since stimulus presentation onset to see when these units were firing relative to the start of the presentation. We can use this to make a raster plot, spike raster for this one stimulus presentation, showing our units of interest from V1 and their spike times relative to the start of this drifting grading presentation. So we also it's also helpful sometimes to bin our spike times. So rather than looking at the individual spike times, we want to know how many spike times occurred within a given window. And so for that, there's a different function called presentation-wise spike counts, which allows you to define the edges of your time bins, as well as the presentation IDs and the units of interest. This returns a X-array data array object, which is similar to a pandas data frame, but allows you to have more than just 2 dimensions, more than just rows and columns. And this allows you to do things like create this binned spiked raster. So for each of these units within a particular time bin, count the number of spikes that occurred. And then you can also average this across trials. So just plotting the using the mean spike counts, basically collapsing across the trial dimension, gives you this kind of 2D map of the responses of all these units to different stimulus presentation, different times relative to the stimulus presentation. So yeah, this is how you would create, for example, a peristimulus time histogram for all of the units in your experiment. All right, so last thing I'll have time for is showing how to do some receptive field mapping. Let me see if this has caught up yet. Yeah, okay, so we're on to our stim table. Our stim table has been generated, so let's go ahead and get the stimulus table for our receptive field mapping stimulus. So this one is called the Gabor stimulus, and we see the length of this table is 3,645. So these are each of the presentations of our localized drifting grading stimulus, which we use for receptive field mapping. We can see that within this table, there are a few different parameters that vary from trial to trial, and we can filter this to figure out the unique orientations, of which there were 3. So basically 3 different directions that the grading was moving, as well as 9 different unique X positions and 9 different Y positions. These drifting gradings also had spatial frequency, temporal frequency, size, and contrast parameters, but these didn't change throughout the experiment, so we can ignore those in our analysis. But to grab the receptive fields for these cells, each stimulus type has a class in the LNSDK associated with it that gives you some convenient methods for performing analysis on that stimulus type. So the receptive field mapping deals with the Gabor stimulus. So if we import this class and then just create an RF mapping object using the session as input, then we can compute the receptive fields for a particular unit or a set of units, you can ignore this warning that that's not important. And so then we can plot the receptive field for this unit. Let's change the color map to gray. And so, yeah, you can now see the region of visual space where this unit was responding most prominently. And then we can do that across a number of different units. And so these were all of our units in V1 that we selected earlier. And now we can see that they have receptive fields basically in the center of the screen. And these receptive fields, compared to ones that you see in the higher visual areas, are quite compact. So yeah, you can use this to look at how receptive field properties change across different areas. You can also use this to look for cells with overlapping receptive fields if you want to look for cells that are likely to have likely to be functionally connected. That's much more likely if they have overlapping receptive fields. So this is just one example of how we can use some of the helper functions within the Allen SDK to compute standard things like receptive fields for this data set. So I have about 8 minutes left, so there's time for questions. If anybody has questions that have come up during the tutorial, happy to answer them now. If there are no questions, I can go on to the final section of the tutorial. But yeah, I'll take a minute or two for people to type their questions if they have them. All right, no questions coming through yet. So I will move to the final section of the tutorial. But yeah, feel free to type in your questions at any time, and I'll pause to answer them. So yeah, last section is just a quick demonstration of how to decode image identity from the neural data. So we can first get our stimulus table for natural scenes presentation. So in this stimulus type, there were a variety of natural images that were shown with a number of repeats. And so we can use a subset of this data to train a classifier that can predict which natural scene is on the screen in the held out data. So we've got our stimulus table that has a frame which indicates the identity of the stimulus that was on the screen, as well as start time, stop time for the interval in which this each image was on the screen. And I think those are the main things that we care about. And so we can pull out all the units in a primary visual cortex by filtering our units table for units in VisP. And then we can use this presentation-wise spike times function that I talked about earlier to grab the spike times for these cells relative to the start of each stimulus. This gives us a spikes data frame with the times of the spikes for each unit. Then you can turn this spikes into a design matrix, which basically gives us, for each stimulus presentation ID and unit combination, how many spikes did that unit fire. So this basically gives us a 2D matrix of with stimulus presentation ID, so the particular stimulus presentation as rows, and the unit IDs as columns. So this is a very large table, and this is what we're going to use to train our classifier. So yeah, the information that we care about is what's in the frame. So which image was actually shown within each trial. And then we can import some functions from the scikit-learn library, which is not actually installed by default through the Allen SDK, so you have to install this separately. And then these functions allow us to split the data into training versus test, and then perform the classification on different combinations of training data. And as you can see, accuracy is around 40%. I guess, yeah, on average, we're getting accuracy of 23% at the end. But so this is basically the probability that the classifier is able to predict the correct image out of 119 total images, in which case chance is equal to 0.008. So even though it's not getting 100% correct, it's still able to do this with reasonably high accuracy. And so we can look at the confusion matrix to see for the actual stimulus ID, what is the probability that the classifier predicted the correct ID. And you can see that the probability is much higher along the diagonal. So for any given stimulus, given just the spike counts across all of the neurons in primary visual cortex, can predict the ID of the stimulus with fairly high reliability. And then this last cell shows how to grab the actual image from the Allen SDK. So we know which of these stimuli was the easiest to decode and the least the hardest to decode. And then we can use the cache get natural scene template function to grab the natural scene from the Allen SDK, and then plot our most decodable image, which is this eagle here, and the least decodable image, which is this field of grass or wheat or something. And so that can be helpful if you want to know what the images actually were. You can also use a similar function to download the natural movies if you want to see what those look like. So that's it for the tutorial. Hope you found this helpful. Again, this exact tutorial is on GitHub in the link that I posted. There's also more tutorials on the Allen SDK Read the Doc site, so feel free to browse through those. And if you get stuck at any point, feel free to post a question on the Allen Brain Map community forum, and someone will get back to you as soon as possible. So yeah, if you have any more questions, I'll stick around for another few minutes to answer them. But if not, I think that concludes this tutorial for the day. And then we will resume again tomorrow with three additional tutorials as part of this workshop.
Yeah, great. Thank you, Josh. Yeah, and thanks everyone for participating. We'll see you tomorrow and talk about cell types, visual behavior, and modeling. And you can see Saskia posted the link to the place where you can post more questions. All right, thanks everyone. We'll see you tomorrow.